Okay, let's go ahead and get started so we can stay on time. We have a packed morning of fantastic speakers. I'm Amy Paller from Northwestern in Chicago. I want to welcome everybody to this wonderful meeting of the International Pachyonychia Congenita Group. And we have our first set of speakers today. I do want to thank Jan and the team for organizing everything. So let's just give her a big round of applause to get started with. And this has been a meeting, of course, that has gone on for many years. And it's great to see a full house here because this is one of my favorite parts I know of the SID, where we just come together to learn about a group of disorders that is near and dear to us, and fortunately is getting some traction and better understanding and new therapies, and that's what this is all about. So it's my pleasure to welcome our first speaker of today, who will talk a little bit more with a welcome address, and that's Pierre Coulombe, who is the G. Carl Huber Professor and Chair, Department of Cell and Developmental Biology at University of Michigan, and he is the scientific leader of research at the IPCC. Welcome, Pierre.
Thank you, Amy. So I'd like to welcome you all to annual symposium of the International Consortium Congenita. I think you'll agree with me that we have a terrific program again this year. Last year was our 20th anniversary symposium. Huge success, and hopefully this year we'll outdo ourselves. The objective of the scientific symposium is to advance the agenda for research that is relevant to Pachyonychia Congenita and relevant conditions, and also to take a hold of where we are in the field and welcome newcomers to join the PC community. I'd like to share that preceding this event this morning, we had a patient-focused meeting on Monday and Tuesday. It was an incredibly successful event, multiple families with people with PCs. They go by PCers of various ages, including young kids. And it was a very powerful and inspiring event. And then yesterday afternoon, we had a meeting of the Medical Scientific Advisory Board, which was very productive. And then we were hosted by Jack Padovano from the Board of Trustees of PC at his home here in San Diego. And that was a lovely evening. Thank you, Jack. So we're very grateful to the PC project, to the Society for Investigative Dermatology for, again, this year allowing us to sort of join the main meeting. I think this is extremely important for us logistically and in terms of impact and recognition. They make space for us in the official program of the SID, and that is much appreciated. I would also like to acknowledge the support of private partners, Biomedics, Kamari Pharma, and 10Bio. Thank you for your support of this meeting. So our program, as you'll see, or you can see from the documents that we shared with you, we have 4 sessions, about 1 hour each. And they are chaired by Amy Paller for this current session from Northwestern, Chris Bunick from Yale, Robin Hickerson from 10Bio, and the last one is going to be chaired by Joyce Tang from Stanford. So I think you can find more information about the chairs and all the speakers through their bios, which are accessible through the QR code that you have on your table. So one of our speakers, Keith Choate from Yale, is, I think, let me see, he's still in the plane. He had some delays last night, and we'll push him down a little bit, but we'll make sure to hear what he has to say. So bear with us as we make those last-minute adjustments. After the second session, we'll have a break, and then we'll try to shoot a group picture. And that group picture is very, very important because we saw at the 20th anniversary symposium last year that when you look at them, you see the beautiful evolution of the project in terms of how many people are in the picture and the additions over the years. That's something that we heard about yesterday, the concept of pacers in PC. Anyway, and then after the last session, we have lunch being served over there. So we'll gather here to have what hopefully will be a robust discussion about, sort of pathophysiology, about how to make us more effective as a group, about therapies, and about anything else on your minds that you feel could propel us forward. I have to say that I've enjoyed a lot of good moments and great opportunities in my own career. But one of the top elements is to have been invited to be part of PC and to do a little bit to try and move this forward. It is incredibly powerful when a PhD scientist such as myself get to interact with physician scientists, physicians, but especially the patients. So without further ado, then I will invite Janice Schwartz to talk about new challenges, new opportunities, and the patients we serve. Janice.
Thank you, Pierre. It was funny when we were putting the program together and we just started outlining things. Pierre put my name right here and he put kind of my title. And it wasn't until, of course, this week that I went, now what's my talk title again? So I guess I'll talk about this. But actually, I will never be not. Two things will always be the truth for me at these kinds of meetings. One is I'm always going to be nervous and I'm scared to death. But two, I'm always going to be just amazed that you are here. You are amazing people. And I even told our MSAD yesterday, I said, I can't believe that I'm sitting at the table with you people. I mean, if you just look, well, I'll show it to you. So without that, let's just get going here. Pierre touched on it, but I just want to do a big thank you. We don't have a lot of drugs in our pipeline, in anybody's pipeline. So we're grateful for these companies that have taken a chance on us and that help us so much. So just a big shout out to these guys again. Oh, yeah. So we are, background. And I apologize for all the people that have seen this slide before, but I always feel like I have to tell everybody about PC for the first time because there's always new people. So just so you know, I am a K6A N171K variant. I didn't know that because I'm old. I was a spontaneous mutation, but I had two children. Dave is here too, and they're his too. And our kids have the typical phenotypes of the nails, the white tongue, the follicular hyperkeratosis in the waistline, of course, the ear pain that so many K6As and others get. But it wasn't until they began to start becoming weight bearing that they got these very painful calluses. And this is really important part of our story is that they are painful. But as they continue to run, they also get traumatized. And so our particular mutation blisters. So not all with PC blister, but they all hurt, but ours blisters. But I was content to do nothing about it because my degree is in MOM. And, but I had a mother-in-law who said, I am not complacent with this and let's do something about it. And those are my 4 kids. They're even older now, but the two on the right have PC. And basically with the help of Sansi Leachman, this is how we all began. And I just always have to say, even though it started with my 2 kids, it really is all kids of all ages and all races and all sexes and all genders all around the world. And we care for them all. And I just wanted to just throw this out. We rebranded a few years ago. I just want to ask you guys, I want to see how you compare to the patients. What does our logo tell you about PC Project? Just shout it out loud. Love. What else? Feet. We're like, we need a logo with feet. We are not a heart disease. But we didn't want to get away from the love. So we got some feet in there. And what else did I hear? DNA. DNA. I know. And they even said, oh, those little dots can be the people. I'm like, okay, that's a stretch, but whatever. We're the people, you know. So the patients came up with all kinds of stuff. But I just wanted to point that out because you see us rebranded all over the place. And wanted you to know that it is a meaningful logo to us. We are loved based unabashedly. We were founded on love. We're still driven by love, but we're not completely fluffy. We are driven by science. We're kind of fluffy a lot, but that's why we brought you, because we're driven by science. So that's where we go. PC project. And again, if you've seen these, I didn't talk about this last year, so I figured I'm okay. Well, maybe I did. I don't know. But we have two major programs. We have the International PC Consortium. That's you guys. That's why we're meeting today. And that is physicians, researchers, and pharma now, because we know we need the pharma people too. We love you. It's okay. And then, of course, our research registry, which is the patients and their data. And together, we think that's a pretty good combination. Why do we need a consortium? Well, because we're rare. Nothing, when we started PC Project, pretty much nothing was being done in our research space. So we needed, I can't see this, brilliant scientists and physicians. We needed collaboration, not competition. And we needed big hearts. And I think we got it. So for the past 20 years, I'm used to reading my own slides. The past 20 years, I'm just going to sit over here. The IPCC has given their love and their expertise. So there you go. And love these people. Last year, as Pierre mentioned, we were all in Dallas. That was excellent. Just wanted to tell you, it's good to see that you keep coming back. Thank you. You also know that if you come, that's why we've got your name on the back on the rolls. If you didn't register, we're going to reel you in anyway. So just know, I just needed to give a shout out to the IPCC steering committee. I think you know a lot of these people. Again, it's a pretty impressive list, and they meet together with me monthly and Holly, because we're kind of a small group, and they advise me, help me, and they're also part of our MSAB. We have a pretty amazing MSAB, and I'll just add that there are 4 of our MSAB members here today. And I'd like to give a special shout out to Chris, who is our newest MSAB member. I don't have his cell phone yet, but I'm sure I will soon. But I pretty much can call on these guys at any time, and believe it or not, they answer. And it's amazing. It's amazing. And I just wanted to let you know, we have this meeting once a year, but the research goes on all year round. I get emails from you guys all year round, and it's fabulous. So if you ever have ideas, don't wait for the annual symposium. You know my number. So I just wanted to tell y'all, thanks for coming along for the ride. If any of you were with us last night, you know what this is. Thank you. And with that, because we are based on collaboration, I have to ask a little favor. Everybody has one of these. Like I said, I'm just a mom. And as you go through the symposium today, I'd like to just invite you to give us feedback. I'm just me. I'm just a person, and I want all the feedback and the help I can get. So as you listen to me talk, as you listen to everybody talk, and you have thoughts, I would very much appreciate it, and even invite you to put your thoughts down and share them. And then when we have lunch, we're going to bring our lunch boxes in here, and you're going to tell us what to do. And you're going to share all the ideas. Sound good? Give me a thumbs up. You're going to do that for me? Good, because I'm just a beggar, so thanks. But I'm very humble at taking advice. Actually, it's like you do with mother-in-laws. You take all advice graciously and then do what you want. That's my plan. So, but we like good advice, right? Okay, so here's our good things. Now we got to go to, why do we have a registry? Because PC, again, is rare. The registry has literally transformed what we know and about PC. And of course, we realize, I'm not going to talk about it because you've got a brochure in front of you, that PC is much more than thick nails. Again, I know I just say this all the time, but if you're new, you have to know that the most common feature of PC is the painful plantar keratoderma. And I was in Amsterdam, and we had this poster at our exhibit table, and a doctor comes up to it, and he looks at it, and he turns to me, and he says, "That's PC." And I said, "Yeah." I said, "Do you have patients that have feet that look like this?" And he says, "Yes." And I said, "Are you testing them?" And he goes, "No." Well, you know how that conversation went. And so I think an important thing for all you to know is that the registry is diagnostic. We are still getting patients of all ages coming to our registry, about 2 to 3 a week, without a diagnosis. And so that's a huge thing. So if you're a physician and if you've got a patient with especially painful PPK, please send them our way. We'll help them. Every test, every case is verified by our genetics team. This is a working genetics team. They get, again, on a call right after the steering committee, and they review your cases, and we get some funky cases. We get some interesting things. So it's kind of cool. Anyway, so I'm going to tell you why I think your PPK patients should receive diagnostic genetic testing. First of all, the registry is life-changing for people. I promise you that people don't want to just have, I call it a nothing diagnosis when they get told they have PPK. There's over 50 genetic PPKs. They want to know why they have the sore feet. They want to know why their skin's the way they are. And once they're with the registry, they have gathered. This was us just 2 days ago, and we take care of them and we love them, and it changes their lives. And you know what? I just got to tell you, we had some, as usual, scientists and physicians with us. We love you to come to our patient support meeting. I'm going to shout out to here Amanda out of Pierre's lab. This is her on the end here. She was so excited because she works on K16 and she saw her very first K16 foot. It was very exciting. So anyway, it was great. But they also really get to see the impact. You wonder why I got a big swimming pool in the picture. When we checked in, we were at a hotel down the street, a lot cheaper, by the way, and we had a view of the pool from everybody had a view, and we checked in, and Dave goes out and looks over the thing, and he goes, there's a PC family down in the pool. And I said, how do you know? And he goes, well, the mom just scooted on her bottom from the from the hot tub to the swimming pool, and she didn't get up and walk. So I went and looked, and sure enough, this is a wonderful family, and we love them. And it's just telling. It's telling. People do not realize the pain and the suffering that these patients have. So, oh well, they just hang out. So we know what to deal with when we have a correct diagnosis. I wanted to just share this story. This happened to me just a month or so ago. This is little Crew. His dermatologist actually looked at him when he was born and thought he had the signs of PC. So she ordered genetic testing, not through PC Project, but from another lab, and got a stupid answer. And it wasn't PC. And the dermatologist was confused, and she said, I don't know, it still looks like PC. But luckily the mom said, well, went looking and found PC Project. So of course she sent me some photos. I said, oh, join the registry, you know. And the reason I have to join the registry is because, like I said, the registry is diagnostic in nature. And so our team reviewed it. He was a K6A. Fast forward about a year, and she calls me one day, and she said, Crew was sleeping and I could hear him, and he just did not sound right with his breathing. We hurried and took him to the doctor, and he just seems to be having problems. Could this be PC? Well, guess what? I immediately, as fast as I could, sent her every publication that we had about the laryngeal involvement that many of you are authors on. And I don't know, they probably would have saved his life anyway. I don't know. But basically, those doctors in hand now knew exactly what was going on with Crew, and they knew what to do and they knew what not to do. And there's Crew. His follow-up appointment was great. So he's doing fine. And I've shared this with her permission. And so it's nice to have a diagnosis. This is a real problem in our community. I didn't realize it was as bad. I think our oldest child is maybe 12 that has had this. It's all the K6As. I'm going to be a little personal right here. Just a little over about 3 weeks ago, I became a grandma. And if that's not the cutest kid you've ever seen in your life. Well, Dave and I were out of the country when that happened, didn't have internet access when he was born. By the time we got home, he'd only been alive for 1 day, and they put a feeding tube down his nose. I'm like, what? And so we get there, and they put him in the NICU. And it's like, why? Well, because he's not eating right. He's not sucking right. So I go in, and I checked him out. I said, do you mind if I look at his nails? And they said, and I looked at my daughter-in-law because he was in the NICU, so it was just the two of us. And I said, I'm pretty sure he's got PC. And she goes, well, he looks perfect to me, which is great. And because that's who she is, because she married my son. And well, he's awesome too, just so you know. I don't want to make it sound like she's settled. He's really cool. Anyway, it was really tricky. And for the next week and a half, that little boy stayed in the NICU, and I about went crazy because he had that tube down his throat. He was a perfectly full-term healthy baby, but they just didn't like the way he was eating. And so they just kept throwing the stuff down and down the tube. Robin was pumping her breast milk, and I even started a hashtag free Benny. But finally, oh yeah, his name is Benjamin David Schwartz, and they call him Benny. So cute. Anyway, sorry, but needless to say, the whole point of this story is at some point I was having acid, and I was just like, anyway, I was just kind of sad that I call and talk to doctors all the time, but because I had the name Schwartz and grandma next to me, I was nothing. But I finally, these guys, I mean, they're not stupid. You know, they got some talking points for me. They went and said, we need to have a consultation with a doctor, and we need to talk about our baby, because they were like, we feel like our babies are being held hostage. And I'm like, he's going to be 25 pounds in the NICU by the time they let him out, because they don't like the way he's eating. And so they're probably wondering why this has to do with research. It's just one of our challenges, but maybe I'm just sharing a little too much. And because we had a cancellation, so Pierre said, talk some more. So here I am. But the long story short, Nate and Robin did great. They got a new person that came around on rounds. She was fabulous, and they believed her. I mean, yeah, no, she believed them. She saw the literature. She saw the things. They knew the pros. They told her about the pros and cons of having that down his throat unnecessarily, and just said he's going to eat funky. So guess what? She pulled the tube out, and he's home. And guess what? He's thriving, as we knew he would, because we know how to feed babies with K6A that have feeding problems. We don't need to stick a tube down. If there is a problem, we'll do it, but we truly don't. So that's them. They're cute. So with that, it was funny because Adele texted me. I told her Benny had been born, and she said, are you going to be one of those crazy grandmas like Mary? You know? And I said, no, I'm already advocating for my own two children. And then I texted her a few days later and said, I changed my mind. I'm worse than Mary. I'm on steroids. So anyway, reason to have a genetic test. Benny's going to be fine because he's got us, and we're going to get good treatments for him anyway. But with that said, we have this other issue going on. People come to us without a diagnosis, and all these people thought they might have PC. They have TRP3, GJB6, K9. Actually, I can diagnose those K9s without a test, but we get DSPs, and they report pain like PC. The AGABs, yeah, some pain, some not. DSGs, again, a spectrum. These guys, even though they don't really have a lot of pain, at least the ones in our registry, they do have that issue with cancer. And so we give them a test and say, hey, talk to your doctor. And you notice one thing, I'm not saying any big names, or am I? We are a gene-based group because we're lazy and we love it. GJB2s, ours just happen to have deafness. I think there's some that don't. And this is really cool. And I think Ellie's going to talk about this a little bit, maybe a lot, because he's doing Adele's talk too. We're really flexible right now. And but we are doing a reclassification. Well, not we are, is done. A reclassification of the PPKs, and we're doing it by their gene names. It's happened, and Ellie will talk about it. But I just want you guys to know what a privilege it was to have a seat at that table. I'm not there because I'm taking the picture, but I promise that was my seat. And then, but we also said, hey, we've been doing this for years, so we're okay. And here we are. These are new names. The one thing is, even though we're trying to get a hold of PC, get rid of PC because it doesn't really define us well, I did kind of talk to my PPK group, and I said, you know, we've been trying for 20 years to get PC on the map. Can we at least be on the end just for a few years till we get rid of it? So here we go. We're official. Thank you, Adele, if you're listening. Welcome to all the Zoom people, by the way. I just wanted to tell you with some of those, these are some of the advice you can give me. How will PC Project embrace all the PEDs that are turning to us for help? That's an issue for us. What do we do with the registry data we're collecting on them? How do we help other peds without losing the focus of our mission? Yeah, we could have 5 hours on this slide alone, couldn't we? And how do we help other peds without sinking ourselves with limited resources? So just something, don't answer now because I'm going to quit in a minute. But but I just wanted to say those are some things that we're thinking about. And if you have ideas, if you're a ped kind of person and you want to give us ideas, write them down. So that'd be great. Or you can email them to me too. It's okay. There are currently no approved treatments for PC. And when I made this slide, I said, gosh, that's boring. And guess what? It's boring that we don't have an approved treatment. So I wanted to say, so we continue on, we carry on. Wanted to just make a fun announcement. PC Project is having a main session in the ESDR in Antwerp for the first time. So if you're coming, come hang out with us. We'd love to have you. So please come to Antwerp if you're going to choose that one instead of the other one. So, okay. Oh, sorry, Chris is, you don't know what you're getting into. We also are part of this is the new reclassification symposium this fall that Anthony, he's on our genetics team, is organizing with a bunch of you, actually, I think. Just some things to keep in mind. And this is breaking news because my MSAB gave us the A-OK for all of you that are associated with this. We are looking ahead at next year. You know how we have this IPC symposium? Next year, we're going to combine. We're not going to have this. We're going to meet with the Derrier people, kind of like we did last year, but we're going to have this amazing 1.5-day meeting and the first ichthyosis group. And so save the date, put it on their calendars because it's going to be epic. And that way, if we hate it, we'll never do it again. So no, I'm just saying that for Corey, who's going to push me for doing it again. I just know it. Okay. Empowering research. Just wanted to let you know that the last 2 years, we've done a grant cycle. And this year, we're going to still do a grant cycle, but we usually open it up here. We're not quite ready. We're tweaking our grant cycle issues. So stay tuned, basically. Stay on the email list so you know when you should apply for a grant. Okay, got that? Okay, money, I know everybody likes that. Oh, I don't have time for this. I just want to say this is one of the things that we're working on is studying. I'm just going to give you full disclosure. I'm on it right now. That's what my feet looked like before ruxolitinib. That's what they look like afterwards. And yet I rolled up here in a wheelchair. Why? Because I'm blistering. And if you noticed me last night after the patient support meeting, I came back to my hotel room and I popped a lot of blisters. I just can't escape the blisters, so they're still crippling me. There's other things too, but basically we got to learn more about it. We got to do a drug. We got to do some off-label studies. I can't see this, so you can read it. You're smart people. And then Kamari Pharma and Biomedics, they're going to talk to you later on in this program. It's very exciting. And I always say this, I show this all the time. It sounds creepy, but I'm like, we know who our patients are. We know where they are, and we know their exact mutation. But the bottom line is it makes us ready for clinical trials. We know how to enroll clinical trials. We're going to need to be strategic about that as well if we get more than one indication. So again, these are our programs for success. We are a family that carries each other. And this is our vision. It's a day when those who suffer from PC will live without excruciating pain, isolation, and embarrassment. And I think the scientists that came this last week to our patient support meeting probably have a better idea now of how impactful and how much that really is for our patients. Want to tell you one concluding little story. I love this story. It's a story of a bunch of people in a church that had to move this big home. I don't know if it was this one, but it was big like this one. And they had to move it from one room to another. And they had lots of people and they could not move it. And finally, one guy just said, let's all stand close together and lift where you stand. And they did it and it worked. And so that is my invitation to this group today. Everybody in here has different expertise. Some of you are here for the very first time. Some of you are long timers, but we all have our strengths. And let's stand close together and lift where we are. Let's be stronger together, lift where we stand, and we can do it. We can move the piano that is PC and everything else. And here we are last night standing together. And we'll continue to stand together. So I'm so glad you're here. Thank you. You're not just the science. I know that. I know you guys are the fluffy part too. We really know you're the fluffy part, and we're grateful for it. So thank you. Thanks again to our sponsors, and let's stay stronger together.
Thanks so much. Wow. What an inspiring start to our session. And congratulations on your new grandson. How wonderful. You gonna move this a little bit? Is this supposed to be plugged into something? Right here. Now my pleasure to introduce our keynote speaker here, who will talk with us about palmoplantar keratodermis, one step forward, and that's Eli Sprecher. I think he's well known to everyone. He's actually now the CEO at the Tel Aviv Sourasky Medical Center. Is it being renamed or not? Not the medical center. Okay. They just got a huge $125 million grant in this past week, so that's great. At any rate, and I think everybody knows that Eli's just been a driver in moving forward everything about keratodermas and what we now call epidermal differentiation disorders, or ED. So I could go on and on, but we're trying to keep our introductions very short to give time for presentations and discussions. So why don't you come on and tell us about one step forward with respect to palmoplantar keratodermis.
Thank you very much, Emily. So good morning, everyone. I'll start perhaps with 2 or 3 thank you. First of all, we are very often hearing you, Jen, telling us how grateful you are for what we are doing, etc. But I think from my personal standpoint, and I think that I speak here on behalf of everybody, it's us who have to thank you for the opportunity to be part of something. And I can very much relate to what Pierre was saying just a few minutes ago, something which to me has been perhaps the most meaningful thing I've been doing professionally speaking. So thank you very, 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 very much.
Сейчас мы вернемся, сейчас мы судим little.
Secondly, unfortunately, Adele O'Toole is not here today, and I will actually give part of the talk she should have been giving today. We wish her, of course, prompt recovery. We are missing her very much. She has been personally for me a long-term partner, and we've done a lot of things together, and we have still a lot of things to do in the future, as you will see in a minute. And finally, I'd like to very much thank you all. Jane was speaking of a family. I really feel that this is a family. I'm very proud to be part of this family and very grateful to each and everyone here for making this PC project such an amazing reality for all of us. So it's really great to be here among a group of fanatic lovers of epidermal differentiation disorders, palmoplantar keratodermas, and in a minute we will rename them, as you will see. So I will start with something which is, I think, very well known to all of us. Palmoplantar keratodermas are clinically extremely heterogeneous, and this is, of course, very much complicating their classification. So for example, it has been customary over the years to distinguish between diffuse and focal keratodermas, between diffuse and punctate keratoderma. But actually the two patients who are represented in the upper part of the slide are, despite the fact that they have distinct forms of keratoderma, are actually carrying the very same mutation in the very same gene, DSG1 encoding desmoglein-1. And the two patients in the lower part of the slides, despite the fact that they were very, very distinct forms of keratodermas, are actually carrying genetic variants in the very same gene, SLRP1. And if this was not enough, it turned out over the past few years that even histopathological features and even genetic data are not strong enough in order to demarcate clinical entities within the palmoplantar keratodermas. For example, let's talk for a minute about epidermolytic ichthyosis, a disorder which is very well known to all of us. So by definition, because this is why it was named epidermolytic ichthyosis, a patient with epidermolytic ichthyosis should display on histopathology epidermolytic changes. But there are now a few reports showing that patients with epidermolytic ichthyosis do not display always epidermolytic changes, even though they are from a genetic point of view affected with epidermolytic ichthyosis. Let's have a look at this patient who has a very rare form of palmoplantar keratoderma called Naegeli-Frances-Jadassohn syndrome. She is carrying an heterozygous variant in the KRT14 gene, which is of course the gene carrying many variants causing epidermolysis bullosa simplex, which share very little in terms of phenotype with Naegeli-Frances-Jadassohn syndrome. So those disorders are not only clinically heterogeneous, they are also genetically heterogeneous, which is even further complication the challenge of classifying these disorders. In addition, many of the terms that we are using in current schemes of classification for palmoplantar keratoderma are actually misnomers, and we cannot avoid the most striking example of all. I owe this to Jen. Pachyonychia congenita, congenital disease of the large nails. But we are all very much aware of the fact that actually the most prominent feature of PC is this very severe and painful palmoplantar keratoderma, with nail changes being variable and of course sometimes only barely visible in some patients. Sometimes the terms that we are using are misleading in another sense. For example, mal de Meleda is not only seen in the island of Meleda, the island of millet, but is actually seen and has been reported all over the world. And finally, some of those nomenclature are actually maybe offending or upsetting for patients. For example, the porcupine people is a term that has been used in order to define patients affected by a keratin-1 associated disorder affecting the tail domain of the protein. But perhaps the most important reason why we really need to update current classification schemes is the fact that we have no ways. I must admit it's really burgeoning attempts, but still a growing number of ways to intervene with the disease course in palmoplantar keratodermas. For example, this palmoplantar keratoderma could have been caused by dominant negative mutations, in which case you would maybe opt for strategies aimed at removing the defective protein, the defective keratin here, from the epidermis. But the very same, morphologically speaking, the very same palmoplantar keratoderma could also have been caused by loss-of-function mutations. And in this case, what is actually needed is some kind of some read-through promoting agent, as exemplified in the successful treatment of PPK Nagashima type. But the very same palmoplantar keratoderma could also have been caused by a gain-of-function mutation. And in that case, maybe you would think of repurposing an existing drug in order to attenuate the deleterious effect of those gain-of-function genetic variants. And this was, of course, very nicely demonstrated in the case of onset syndrome and treatment of onset syndrome patients with erlotinib. So a lot of challenges. In order to meet those challenges, a group of 19 experts convened over a period of approximately 2 years in order to define a new classification scheme or disorder of epidermal differentiation. This classification scheme has now been summarized in 4 papers, which were recently accepted for publication in the British Journal of Dermatology, and they are now available online. And there are 3 principles behind this new classification scheme. First of all, we are getting rid of previous unifying terms like disorders of keratinization and disorder of cornification and MEDOX and etc. And we are adopting one unifying term to define all disorders affecting epidermal differentiation disorders, or EDDs. And there are 3 major subgroups of EDDs. One are non-syndromic EDDs, or N-EDDs. The second one is syndromic EDDs, or S-EDDs. And finally, there are EDDs which are more prominently involving the palmoplantar skin surface, and those ones are called P-EDDs. Secondly, the new nomenclature is aimed at emphasizing etiology instead of morphology. And each disorder is named based on a combination of 3 different elements: the gene, the type of EDD, so N, S, or P-EDD, and finally a clinical descriptor wherever needed. So for example, ichthyosis vulgaris is now called FLG-N-EDD, and epidermolytic PPK is now called KRT9-E-EDD. And a more complex one, Reim-Munk syndrome, is called CTSC, the cathepsin C, encoding gene, PEDD, and then arachnodactyly in order to distinguish this very rare syndrome from the more common Papillon-Lefebvre syndrome. So wherever there is a need for some kind of fine-tuning of the classification, then we add a clinical descriptor. And thirdly, the different forms of palmoplantar keratodermas, now called PEDDs, are grouped into 5 major functional groups, and each of those categories is actually pointing to one defective functional element: structural proteins, enzymes, channels, transcription factors, and signaling molecules. And in each of the 3 papers describing each of the 3 major subgroups of PPK, you have tables which are explaining why and how are these different forms of epidermal differentiation disorders belonging to a certain functional group. And in order to ease the transition from the previous classification to the new classification, each paper is also providing a diagnostic algorithm, which is not only connecting morphology to etiology and thereby helping the clinician when faced with a challenging clinical case, but it is also demonstrating how easy it is now to incorporate new entities, and there will be new entities as they emerge, not only in the classification itself, but also in the diagnostic process. And I'd like to give you a short example to try to illustrate this point. So we have been speaking about DSG1, and we, of course, know that heterozygous and sometimes biallelic variants in the DSG1 gene are associated with various forms of palmoplantar keratoderma. It was a group of Cathy Green who several years ago demonstrated that desmoglein 1 is not only a very important adhesion protein, but is also a very important regulatory protein, which is regulating some aspects of epidermal differentiation in cooperation with several signaling molecules, regulating the activity of ERKs. And when ERK activity is increased, you get tissue hyperplasia, and in our case, palmoplantar keratoderma. Now, independently, it turned out that a number of palmoplantar keratodermas are also associated with genetic variants affecting either the expression or the function of protease inhibitors, such as, for example, serpin B7 in the case of Nagashima type PPK, and serpin B8 in the case of exfoliative ichthyosis, and serpin A12 in the case of a recently described autosomal recessive form of PPK, and CSTA, encoding cystatin A, in the case of peeling skin syndrome type 4. So those protease inhibitors were deficient are associated, of course, with increased proteolytic activity within the epidermis. So for example, in the case of serpin A12 deficiency, you get an increase in the activity of kallikrein 7, which is, of course, leading to degradation of their substrates. In this case, desmoglein-1. And we mentioned the fact that desmoglein-1 deficiency is associated with keratoderma, and corneodesmosine, which has been found to be associated with different types of peeling skin syndromes. And this is nicely shown here on Western blot. So why is this important? Again, to illustrate the connection, the intimate connection between morphology and etiology. This is connected to the fact that patients with keratodermas associated with genetic variants in genes encoding protease inhibitors usually will display a double phenotype. On the one hand, keratodermas, as we mentioned before, very often, maybe due to the fact that those genetic variants are associated with desmoglein-1 deficiency, and peeling, very subtle but still always present peeling of the skin, which may be associated with the lack of corneodesmosine expression due to abnormal activity of those protease inhibitors. Now, similarly, genetic variants in the KLF4 gene have been shown to be associated with a diffuse form of palmoplantar keratoderma. KLF4 was shown to induce the expression of desmoglein-1. And quite interestingly, patients with KLF4 mutation have on histology widening of the intercellular spaces. And this is really a kind of a hallmark histopathological feature of all keratodermas associated with desmoglein-1 deficiency, whether directly or due to germline mutation in desmoglein-1 or indirectly. And in parallel, there are a number of keratodermas which have been associated with increased EGFR activity and leading to increased ERK activities, such as, for example, Pachyonychia Congenita, and this was the work of Anna Ovnenian, due to genetic variants in different keratin genes, the onset syndrome associated with TRPV3 genetic variants, and a more recently reported keratoderma associated with mutation in the gene encoding cathepsin C. So clearly, this actually demarcates a subgroup within the subgroup of keratodermas that we are describing here, which of course belong or connect all of these to the very same pathophysiological pathway. More recently, we identified semi-dominant mutation in a number of individuals affected by a form of PEDD of very, very variable severity. Some patients were severely affected, others were only mildly affected. And those semi-dominant mutations were found in a gene called DUSP1. And the mutations were found to lead to decreased expression of DUSP1 as well as the phosphorylated form of DUSP1. DUSP1 is a known inhibitor of ERK, and as expected, when those PEDD-causing variants were introduced into keratinocytes, ERK activation was noticed, as you can see in here. But what is more important, or perhaps more interesting, is the fact that those variants were also associated with decreased expression of desmoglein-1 at the cell membrane in keratinocytes. And this actually suggests the existence of a kind of a self-reinforcing pathophysiological loop, which may underlie the pathogenesis of this new form of keratoderma. When the skin equivalents were generated with DUSP1 deficient cells, you can appreciate here the existence of the generation of hyperkeratosis and acanthosis, which is very much reminiscent of what we saw in the patients. But again, what is quite interesting here is to see the desmoglein-1 expression at the cell membrane of the keratinocytes was very much reduced as well. And accordingly, when we use the dispace dissociation assay, which is measuring the strength of cell-to-cell adhesion in epidermal sheets in vitro, introduction of the PEDD causing variants was associated with decreased cell-to-cell adhesion. And this abnormal phenotype could be completely rescued using an ERK inhibitor, as shown here by the dispace dissociation assay. And again, completely in line with those data, when we repeated the same experiment in keratinocytes, introduction of the two variants was found to decrease the expression of desmoglein-1, but desmoglein-1 expression was rescued completely using an ERK inhibitor. So in short, those semi-dominant variants in DUSP1 cause a new form of desmoglein-1 related PEDD. This is in itself interesting, but I was asked to speak here mostly because of the new classification. So let's go back to the new classification. And I think that looking at all those data, it is quite clear that it is not only very easy to name new entities as they come up, for example, in this case, DUSP1 PEDD, but it is also actually very easy to incorporate them not only into the existing classification, but actually into the diagnostic algorithm. From a clinical point of view, this is, of course, extremely useful. More importantly, the demarcation of these functional subgroups is, of course, pointing at novel therapeutic targets, and there are here a number of druggable targets, such as ERK and EGFR, and of course, the kallikrein. So in short, I think that this is perhaps now time to revisit our overall general approach to disease classification and to epidermal differentiation disorder classification, more specifically to move away from morphology and symptom-based therapy to mechanics and pathogenesis-based approaches to therapy for those conditions. And just before closing, I'd like to very much thank all the people who have been part of some studies that I presented here, especially the EDD classification team, or collaborators in Israel and abroad, who work with us on the DUSP1 story, especially Kirill Malovitsky and Ofer Sarig. And I'd like to very much also acknowledge our funding agencies on this project. And thank you very much for your kind attention. Thank you.
I think this is a great opportunity to open it up for anyone who has questions about any of the research that Ellie presented, or the new classification, which we are pretty excited about, but understand may have some controversy related to it. So let's open it up if anybody has anything to ask. Yeah, go ahead.
Congratulations for this, huge contribution. I have a, really forward-thinking question. So we work on a gene that has name changed 5 times. First, it's chromosome open reading frame 86. Then based on, cloning oxygen drosophila has one name. Then based on protein structure has another name. Then a complex fourth name. Now the molecular pathway, that's actually where you're going. So, so what do you think this naming will change, in the future? As we know, science is, professional.
So difficult to say where I mean, we are still actually in the process of trying to change things. So difficult to speculate about how things are going to change will further change in the future. But I think that what is extremely important here, beyond the nomenclature, is to try to realize why is this new classification needed. And as clinicians, we are more and more often faced with situation in other fields where the names are actually used in order to guide treatment choices. So I think that the field which is most nicely exemplifying that is oncology, where I will perhaps speak about a little bit in an exaggerated fashion, but you don't care what is the origin of the tumor. It can be in the lung, in the stomach, or what is important, what is the pathway which is altered by the driver mutations. And that's the pathway that you are going to target. So the same rationale actually underlies our attempt at reclassifying those diseases. And naming a disease by the gene name is actually somehow pointing the clinician to the pathway that should be targeted.
So for one particular disease, would there be 2 different groups claiming the same disease with different names, depending on what they're focusing on?
Well, the question again is what's in the disease name. So if you speak about the disease as it was named in the past, you may be right. But from our standpoint, there will be just one name per one disease, because one disease should be named according to its etiology, to its underlying etiology, not according to its manifestations.
I looked at that algorithm and my eyes crossed. Is there some enterprising medical student or researcher that wants to make a nice app so that us clinicians can put in our features and have it spit out the correct name at the end?
It's in the making. Yeah, entirely right, but it's in the making.
But so you should also look at these papers that have come out because they're really designed to help clinicians to, with huge tables that give great detail about the clinical features that tie to these names.
Great talk as always, and I was excited to hear you talking about ERK and MEK and thinking about some of our own work that's pointing to these same pathways that may underlie different disorders that kind of converge on a mechanosensing differentiation pathway. But I'm wondering, maybe we can talk about this in the strategy session. How do we get traction with the companies that own those drugs to think about repurposing them for these rare disorders to potentially help patients? I think that's something that is an obvious thing for me that I think we need to test. And it's just how do we get over that bridge to the companies to get them to think about PC or other keratodermas to move it toward patients?
Yeah, so I think that maybe we should keep this question for the strategy session. Yesterday we were actually we raised this issue of the traction issue, and on the one hand we have the patient, the industry has the way to develop, or in some case to repurpose or donate some of their compounds to be repurposed for those rare disorders. And I think that the key here is how do we unite as clinicians and perhaps condition or consent to participate to some clinical trials for rare and perhaps lucrative projects to the consent of the industry to also dedicate some of their resources to the study and treatment of rare disorders. So maybe that's a general idea.
We're 3 minutes ahead of schedule. Please take this 3 minutes to fill out section 1 of that form, feedback for PC project. And I'll use a couple minutes to kind of talk about my road with PC project and the FIRST Foundation. So I am very honored to be a part of the medical science and advisory board for both PC Project and the FIRST Foundation. And it's a journey that's been 10 years in the making. And I want to give a shout out to Dr. Lynn Millstone. I don't think he's here in person. I hope he's here online. But Lynn Millstone was my dermatology mentor at Yale. And he really encouraged me in my early years of working on the structural biology of keratins to get involved in PC Project and FIRST Foundation. And these two foundations really sparked my young career. I wouldn't, I think, be anywhere as a physician scientist without these two foundations. And it turns out I happen to be editor-in-chief of Dermatology Times right now, and I have to write a monthly column. And I wrote my column this month on PC Project and FIRST Foundation and how it transformed my career and to try to mobilize dermatology, both practicing dermatologists, researchers, and industry, Corey, to come together and help rare genetic disease. So please read that article and encourage your colleagues to get involved. Money-wise, that would be great. Personal-wise, time-wise, that's okay too. And, you know, Janice, what you were saying about Benny, I mean, I was tearing up here because in the end, it's all about the patients. We are all here for the patients. And as a physician scientist, I can tell you one of the greatest joys of my career in the last couple of years has been as a principal investigator on the clinical trials for a drug for PC and for ichthyosis. Unfortunately, neither drug is going to get approved for these diseases. But what keeps me going every day is the hope that those patients had that these drugs would work. And I am telling you, these patients are craving therapies, and it is our job. I really believe it's our job here to keep fighting for those therapies. And not every therapy is going to work for every patient. So we have a long road ahead of us, but I can tell you that that mission is incredibly important, and the patients are looking to us as, I don't want to say as heroes, but as the people that are going to give them and sustain that hope. So I'm really excited to chair the second session on advances in genetics and disease mechanisms with relevance to PC. And our first speaker is going to be Dr. Benjamin Naines. He's an assistant professor of dermatology and bioinformatics at UT Southwestern Medical Center. And he's been really pioneering some research on the non-mechanical functions of intermediate filaments. So we're going to hear from Ben, and just for everyone, Keith Choate's plane did make it, but I am going to have him last in the session to give him time to collect his thoughts. So Ben, the floor is yours.
All right, well, thank you. So this is the keratin intermediate filament cytoskeleton in a keratinocyte. And, I think looking at this depiction, it's easy to imagine how these protein polymers form a scaffold that allows the cell and its tissue, the epidermis, to resist mechanical force. So it's a mechanical or structural protein, right? But one of the quirks of the intermediate filament cytoskeleton is the sheer number of different proteins, or in the case of epithelial cells, different keratin proteins that can construct it. So there's 54 keratins in the human genome. They're expressed in different combinations. And this has, over time, led to a lot of interest in potential non-mechanical or other functions that individual keratins might have in addition to their shared mechanical function. But this turns out to be very difficult to separate out. And here's an illustration of why. So these two cells are labeled for keratin 5, but what is different between the two cells is keratin 1. So the cell on the left has an amino acid substitution in keratin 1. This happens to be a substitution that can cause epidermolytic ichthyosis, but if you make the equivalent substitution in keratin 6, it can cause PC, or the equivalent substitution in keratin 5 can cause EB simplex. But at the cellular level, what we see is more or less the same thing, which is disruption of the keratin filament cytoskeleton, right? And so the problem is, if there's something that's different in this cell, it's hard to say that it is specifically due to a loss of keratin 1, when what we have really done is disrupt the entire keratin filament cytoskeleton. And this is true to some extent with any loss of function model, including a knockout that depletes enough keratin that the cytoskeleton becomes fragile and there's loss of cell and tissue stability, which can have very broad effects and make teasing out a function that an individual keratin might have that might be unique very, very difficult. So we wanted to try to untangle this in the setting of skin wound healing, which notably is associated with an increased expression of the same keratin isoforms that are implicated in PC. So rather than depleting keratins, we thought we'd try the opposite approach. We increased expression of individual keratins, but in a controlled way, so we could tune the mixture of keratin isoforms in the cytoskeleton without disrupting the overall filament network or mechanical function. We made keratinocyte cell lines with either a little bit more of keratin 6A, one of the PC or wound-associated keratins, or a little bit more of keratin 5, a closely related steady-state basal epidermal keratin. We grew these cell lines into organoids, created wounds, and what we saw is that the organoids with more keratin 6 would close a gap faster than organoids with more keratin 5, right? Usually we think about epidermal remodeling inducing expression of keratin 6. What we see here, though, is that engineered increased expression of keratin 6 induces epidermal remodeling behavior. So it's a causal relationship here. Now, as far as we could tell, there was no difference in the ability of these cells to resist mechanical force, right, the structural role of these proteins. What we actually saw was that cells with more keratin 6 generated more contractile force. Now, this is not keratins directly, this is actomyosin contractility, and we could show that there's increased myosin activation through a phosphorylation switch on myosin regulatory light chain. So this would seem to be a context where we, in fact, can separate out a structural or mechanical and a non-mechanical function of keratins that depend on the specific isoform. But how might that work? So there have been a couple paradigms proposed previously that could apply here, including keratin filaments binding and stabilizing other proteins or affecting their subcellular localization. But what I want to suggest is something a little bit more subtle, which is that proteins interacting with a filament, a one-dimensional structure, are positioned so that they are much more likely to interact with other proteins that interact with that filament, right? It's sliding around in one dimension versus floating in a three-dimensional cytoplasm. But, regardless, for any of those paradigms to apply here, there have to be proteins that differentially interact with keratin filaments depending on their isoform composition. So that's what we wanted to look for. We, of course, had to do this in a way that didn't actually disrupt the filament network. And so we used a strategy of proximity biotin labeling and proteomics. Basically, this allows us to identify all the proteins near keratin filaments and directly compare cells with more keratin 6 to those with more keratin 5. And I'll point out, first of all, we did identify myosin subunits associated with keratins. This has been reported before, including by Pierre's lab. But what's important here is that this does not depend on the isoform mixture. So by itself, it's not sufficient to explain why we see increased myosin activation in cells with more keratin 6. We did identify a number of proteins that were differentially associated with keratin filaments depending on the isoform mixture. One of these really stands out, and that's vimentin. Now, vimentin stands out because it is also an intermediate filament protein, but it's the principal intermediate filament of mesenchymal cells, not epithelial cells like keratinocytes. So already this is a little bit weird. Now, it's not crazy to think that epithelial cells in culture would express some vimentin. That is often the case. And, in vivo, this certainly happens during cancer development. You can see co-expression of keratin 6 and vimentin and squamous cell carcinomas. And so it may be that this particular interaction is mainly relevant in that context, but I do want to delve into it a little bit more because of what it tells us about the biology of intermediate filaments. So the other interesting point here is that we don't actually see vimentin filaments in these cells, right? We see a diffusely cytoplasmic immunofluorescence pattern. About 1 in 1,000 cells have small aggregates or squiggles of vimentin. But this really is a diffusely cytoplasmic pool, right? We can verify this biochemically, and we can actually detect small oligomers of vimentin in these cells, including tetramers, which is one of the more stable states of intermediate filaments in vitro. We don't think this is simply because of low abundance of vimentin in these cells. We think that vimentin is arrested in the small oligomeric state because it's post-translationally modified, including through phosphorylation and citrullination. And these are both modifications that in other contexts have been shown to disrupt vimentin filaments in the case of phosphorylation or prevent complete assembly in the case of citrullination. So, in this model, we do see co-expression of keratin filaments and a small soluble pool of vimentin. They interact. That interaction is increased when there's more keratin 6. We can confirm this biochemically by cell fractionation. We can confirm it in situ using a proximity ligation assay. And we can confirm it in vitro as well. So this is a collaboration with Ohad Medalia's group. They recombinantly polymerized keratin filaments containing either keratin 5, keratin 6, or a chimeric keratin containing the head domain of keratin 6 attached to the rest of keratin 5. They mixed these with unpolymerized vimentin and measured the interaction. And the interaction is increased when the keratin filament has at least the head domain of keratin 6, right? So it's a direct interaction, it's specific, and it is the head domain that is mediating the isoform specificity. Okay, so remember I mentioned so this is now getting a little bit away from myosin activation, but the way we can get back to that is because vimentin is phosphorylated. And so that means that vimentin interacts with kinases, including kinases like ROCK and MLCK, which can also phosphorylate myosin regulatory light chain. And so we could potentially put together a model like this. When there's more keratin 6 in the filament, there's more vimentin recruited to that filament, and maybe it shuttles one of these regulatory kinases with it, organizing it in a geometry where it is more likely to interact with myosin subunits, which are also interacting with keratin filaments, which could potentiate light chain phosphorylation, myosin activation, and increased cell contractility. So if this is right, we should be able to push on vimentin to influence this pathway. And that's exactly what we can do. If we increase expression of vimentin in keratinocytes, we see increased cell contractility. And conversely, if we deplete vimentin, we see decreased contractility. We can bring down the level of light chain phosphorylation in cells with more keratin 6 back to the level we see in cells with more keratin 5. So the way that we're getting from a change in composition of the ostensibly mechanical keratin filament cytoskeleton to this phosphorylation event, the signaling event, is through a combination of isoform-specific interactions with keratin filaments, dimensionality reduction to organize interactions, and very interestingly, through pools of soluble non-filamentous intermediate filament proteins. So what does this mean for PC? I mean, I think, first of all, I think it's not that, I think it makes sense that a medication that might inhibit a signaling function might decrease inflammation, might not be able to rescue the fragility caused by mutations which disrupt keratin filaments, and might not be able to prevent blistering. And I think so we think about therapeutic mechanisms, we need to think about both. And then, the flip side of this is that in some respects, PC is related to other keratinopathies with very similar mutations that prevent keratin filament polymerization. And maybe, with the right therapeutic approach, there could be one therapy that could target a number of rare diseases together. And that might be something that is easier to actually test and get FDA approval for. So I'll stop there. I want to thank two people from my lab who did a lot of the work I showed, Sabahat and Surbi. And I also want to thank the Pachyonychia Congenita project and FIRST, both of which supported this research. So as far as we can tell, no. I will say that the depletion of I mentioned that I showed was in cells that we engineered to express more keratin 6. So that may not, I can't say it wouldn't change it at all without that forced expression.
Great talk. I was wondering whether there's a way for you to test and see whether this interaction changes with like a stress condition, mechanical stress versus a heat, and whether that alternates how this interaction occurs, and this is something that can be targeted in the physiological or pathological condition.
Yeah, that's a very good question. Right now, this is a difficult interaction to detect. We largely are using culture-based models. I think there are ways we could apply mechanical stress to these cells, like at least stretching, which is not a perfect analogy for what happens in vivo, but that's something we could certainly test and see if that affects which proteins interact with keratin filaments. Yeah.
Last two questions, Nancy, and then Franklin about.
So I just wanted to kind of a follow on is the contractility. What are you actually proposing, or have you thought about how that actually functionally relates to, say, the blistering and things like that?
I'm not sure that it does. I think the blistering is related to the fragility of the keratin filament. So these mutations in the conserved portions of the rod domain basically interfere with various stages of oligomerization of intermediate filament proteins. And that's why at the cellular level, you see basically the same thing, whether you express keratin 1 with that substitution versus keratin 6 with that substitution. Yeah, so we have not. Now, I think it's very interesting that the cell might have a mechanism to tie control of a pathway that's going to generate internal stress with the structure that's required to ensure cell stability under that stress. Whether this is relevant in PC specifically versus cancer, that I don't know. Yeah.
So I have a question. Bear with me. It'll be a quite thick question. So related to the PC, we know that there's mutation on keratin 6A, right? And then when you were talking about soluble vimentin, with the contraction, right? So in such a way, if we eliminate the soluble vimentin, would it mean that with a huge amount expression of the mutated keratin 6A in the PC case, would it mean that it would somehow change the blistering?
I don't know. I don't think so.
Because what you were mentioning is that right now we have in the PC case the wrong mutation of keratin 6A. Right. But then there's still soluble vimentin in right now, which increasing the contraction and kind of like try to keep, and the cells still try to maintain the normal physiologic, but because of mutation 6 of keratin 6A made wrong, then kind of like the blistering is my form and the hyperkeratination is happening. But after, if you actually eliminate the soluble vimentin, which reduce the contraction, and that might change a different course. So that's why I was questioning, if you target the soluble vimentin, would it be a case?
I think the connection between myosin activation and cell contractility and more complex tissue behaviors is not always straightforward. I think what is clear is that these mutations cause a very clear structural defect in the keratin cytoskeleton. That may be entirely, so it may be that this particular interaction plays some role in the transformation of cells during squamous cell carcinoma, right? It may even be that this pathway is activated in anatomic sites where there's more keratin 6 expression constitutively, like palmoplantar epidermis. But it may also be that this pathway has nothing to do with the pathophysiology of PC, which might be driven more by the structural defect in keratin assembly caused by the mutation. So both of these things could be on separate tracks. I think, what I would argue is that we need to actually try to separate these out, because right now we don't really know. Thank you.
So next, we're going to have Dr. Corey Simpson. He's assistant professor of dermatology at the University of Washington. Come up and talk to us about a human epidermal model of keratin 5 loss of function and how it reveals potential therapeutic strategies for epidermolysis bullosa simplex.
So use a roving mic, is that okay? Thank you. Okay, wonderful. I have to be able to see these slides to give the talk. So my name is Corey Simpson, and I'm at University of Washington. I'm very excited to be here. I've been talking to Janice for a number of years. We met actually on Zoom during the pandemic when I had nothing else to do. And it's like, yeah, come on. I don't know about PC, but sure, join. And it was so welcoming, and I'm so thankful to be here. And we finally actually have a keratin related project. So I was super excited to be able to present this. So my lab really tries to understand how we get from a patient like that shown on the left with rare blistering diseases to understand the underlying pathophysiology of those diseases, both at the level of the tissues that fall apart, the cells that make up those tissues and the bones of those cells, their adhesive junctions, and as well as the actual molecular structures that hold these cells together, like desmosomes shown there on the right. And this is a beautiful desmosome electron micrograph, but that's a very static image of what that junction looks like. And now what we can do is harness the power of live imaging to bring those desmosomes to life and allow us to watch how keratinocytes adhere and behave in an in vitro tissue that can actually then hopefully inform the way that we treat and understand these diseases. So these are my conflicts of interest. On the next slide, we do have a patent on the use of MEK inhibitors for skin fragility disorders. I've done a little bit of consulting to help to move some of these therapies toward therapeutics. This is my lab. We are based in South Lake Union. Here's the needle here. So these are the wonderful people who have taken a chance on a very new lab and have come to join me to do some research on rare diseases. So I'm very thankful for all of them for taking a chance on me. I want to give a first shout out to NIH-funded basic science, because I think this is really critical, and I'm willing to take part of my time to talk about this. I started in a fruit fly lab. What did that have to do with patients and medicine? I didn't know at the beginning, but now I understand how important it is to have really critical basic science model organisms so that we can understand complex diseases. I next went into Kathy Green's lab where she was focused on a very critical organelle that translates into defects in the epidermis that cause blistering diseases. And then finally, I learned how to do live imaging from someone who was doing very basic neuronal cell biology, and I was able to apply it to the skin. The reason I bring this up is because all of these would not have happened, and I would not be here today if it were not for investment by the National Institutes of Health to fund my training and the research that has allowed me to get to where I am today. So I just want to make that unabashed endorsement of the NIH during a time when I think they're being threatened. And this is really the lifeblood of medical research for patients. So I just wanted to make that absolutely clear. I study keratinocytes, as many of the people in this room do, and these are hooked together by desmosomes, which are linked to the intermediate filaments. These are really critical, and we know this from diseases in which these cellular structures are compromised. This leads to blistering within the epidermis, and mostly my clinic focuses on a disorder like this, where the spaces are between the keratinocytes. This is pemphigus. These can also happen in a family of genetic disorders, and I went to Penn to learn how to treat these disorders as a dermatology resident, and these two wonderful physician scientists started referring all of their genetic blistering disease patients to me because we really didn't have great therapies for them. So there were wonderful treatments for autoimmune versions of these disorders like rituximab, but they didn't have anything for these genetic blistering disorders, which is the same as the case for PC. So this really stood out to me as like a major hole in our understanding of how do we help patients who have genetic blistering diseases that deal with this from birth all throughout life. So we've recently been studying epidermolysis bullosus simplex. This is something that is an onset at birth, causes skin fragility, painful wounds, can also affect the mucosal tissues in some cases. It tends to improve with age, which is interesting, but they still get trauma-related blistering, but they also get palmoplantar thickening, which I think is interesting. And this tends to be caused by fragility of the basal keratinocytes. And this really important paper that came out, and the author is sitting here in the audience, really helped to link these genetic mutations in keratin 5 and 14 basal keratins to these phenotypes to help us understand that actually the really important role of keratin dimers in providing structural integrity to these tissues. So this can actually be inherited in an autosomal dominant way, but it can also, there have been case reports of autosomal recessive versions in which patients are effectively knockout for either of these genes, suggesting that it may not only be functioning in a dominant negative version, but it could also be just the loss of these proteins can disrupt the balance of keratins in the cells. But despite knowing the genetic cause for over 30 years, there are still 0 FDA-approved drugs for EB simplex. There are some new approvals for dystrophic EB, which has been very exciting. I think the limitations of those are real, but at least there's something that's great, but there's really nothing for many of these patients. I pulled this original, one of the earlier publications because Jenna Seibert is actually in our department and recently retired. But I noticed that in one of these reports of EB simplex, they actually showed the feet. And this reminded me of a lot of the pictures that Jana showed. And then I had one of my own patients that came in, and her main complaint was that she had these very painful calluses, but she has a keratin 5 mutation. So I think that's very interesting, and just trying to make a connection with the PC group here. This is the original animal model of knocking out this gene. So keratin 5 is gone here. But what was interesting to me, this was in 2001, is that there was an upregulation of keratin 6. So there is crosstalk between these keratin filaments, and there is some sort of regulation that supersedes any individual keratin. I think Ben was getting at this too. The ratio of these things and how they react with one another and cross-regulate each other may actually determine the phenotype of these disorders. So when you have a perinatal lethal mouse, it kind of stops some things. It's very difficult to do fundamental basic science research on mice that die perinatally. It's hard to sort of keep this going. And so we asked, well, can we replicate EB simplex in a human tissue model, which I've been using in my lab to study some other blistering disorders. So this is where Karina Schmidt, who's a very talented PhD student who's here in the audience, has brought this project forward. It started with a collaboration with Johan Johnson's lab at the University of Michigan to create CRISPR-generated knockouts for keratin 5. Karina's going to talk a bit more about this project in the mini symposium on Friday at 9:30 on epidermal structure. But we were able to engineer keratin 5 deficient cells. We got really efficient knockout, which is a great start. And then what Karina did was to look at, well, how does this affect the overall intermediate filament cytoskeleton of these cells? And what you can see is that when we lose keratin 5 in the two right-hand images, the keratin filaments look disordered. They collapse around the nucleus. They probably aggregate in some ways. And this is just from removing. This is not from inserting a mutation. It's just taking away 1/2 of a dimer that should be present in these basal keratinocytes. We know that keratins are really important for linking desmosomes. And so we asked whether they had impaired integrity if we grow them as cell sheets. And so this assay has already been mentioned by Dr. Sprecher earlier, but we see these keratinocytes and then lift them up as intact sheets, subject them to mechanical stress to basically see how well do they hold up to shear stress. And so what we noticed was that if we apply mechanical stress, the keratin 5 null keratinocyte sheets actually fragmented quite a bit more than the controls. And so that may not be that novel of a finding, but it gives us a somewhat scalable system by which we could potentially screen for pharmaceutical compounds that could actually turn this phenotype around in a more efficient way than trying to treat knockout mice or trying to do sort of a human intervention before we really know whether the drug could work in vitro. And I think this could be something that could be scaled for pharmaceutical screening. So we actually took this into an organotypic epidermal model to see if we could make a tissue that looks like EB simplex. And so when we put keratinocytes at an air-liquid interface, they do their thing and stratify into multiple layers. You can see in the control cells, they form a nice, actually pretty well cornified epithelium. But when we remove keratin 5 from this system, you'll see something interesting. One, we actually see the basal layer cytolysis, which is similar to what you would see in a patient. But interestingly, the cell seems to hyperdifferentiate. So this remarkably looks like the cytolysis seen in the lower layers in a biopsy. But what's also interesting is that you get these other layers that actually escape the basal layer and immediately flatten, which suggests that there may be a way to escape that basal phenotype to allow them to actually form these upper layers that may be able to perform a compensatory role with the skin barrier function. And so that's really what we're looking at. Can we use this model to find new treatments to basically screen it in a high throughput way and then apply it to a model like this and say, is there hope for patients that have this type of a mutation? Could we actually use this system to screen for something that can fix the histology? And would that give us a better idea of what might actually work in a patient? So this is what the keratinocytes look like. And these are some beautiful images that Karina has taken. What we notice is that in our knockout cells, they actually start to express keratin 1 and 10 earlier than they probably should. And they actually flatten out into these big pancake-like cells as sort of a reaction to not having basal keratins. We think they actually turn on epidermal differentiation pathways prematurely, which is an interesting thing. And they actually seem to adopt more of a granular layer phenotype earlier than they probably should. So it seems like by lacking keratin 5, we've sort of shrunk down the epidermis, which is why they were sort of diminutive looking, and kind of rapidly go from a basal phenotype into a granular layer. And here we use filaggrin as a marker of that granular layer. And we can see that they increase the expression of filaggrin earlier than they would in a normal situation. So I think that's interesting and actually makes for a potential way that if we could swap in different keratins, maybe there could be actually a therapeutic in this type of a result. To think about ways to upregulate these keratins, could that actually be compensatory for cells that don't have a good complement of the basal keratins? So the interesting thing was most of our knockouts looked like this, as I showed you before, with the basal layer fragility and then these flattening of the upper layers. But occasionally, we got a couple of knockouts that seem to have a different phenotype. They still have no keratin 5, so they're completely knocked out. But we called them comp lines because they were actually able to differentiate and almost hyper-differentiate. They seem to have this sort of hyperkeratotic phenotype where they're thickened. And this can actually happen in patients where they get hyperkeratotic or hyper-thickened areas, especially in areas of high mechanical stress. Could this actually be a way to compensate for the lack of keratin 5? So we're really using this platform to try to understand, could we find out downstream pathways? If we can't fix the keratin itself, is there something downstream of this loss of function that could be used to halt skin blistering? So that's something we're very excited about. We've started to do some RNA sequencing to sort of take an unbiased approach at looking at pathways that could be druggable, things that could actually be brought to a patient. I'm very optimistic about CRISPR and gene replacement, gene editing, but I think for right now, the quicker way is actually to find actionable pathways downstream of these mutations that could actually be intervened upon now with existing drugs and not have to engineer a totally new viral delivery platform, gene replacement platform, CRISPR editing platform that really hasn't made it, we've had a couple make it through clinical trials, but it's a big lift to design a brand new vector for each individual mutation. That's going to be really probably a ways in the future. So I think this is really exciting to think about ways that we could target these. I just want to give a shout out that this model has worked. We did this for dairy eye disease where we took out here circa 2. And we actually saw that there was a decrease in keratin 10. We screened for ways to improve the phenotype and found that the MAP kinase pathway was actually hyperactivated, which is what Dr. Sprecher was talking about just a few minutes ago, and actually found that if we intervened on this pathway with a molecular inhibitor that's already approved for other disorders, we used a MEK inhibitor and could rescue the expression of keratin 10 in these keratinocytes. And more importantly, it allowed them to actually stick together better. So if you're looking at a patient who gets trauma-induced blistering, if you could do anything to actually increase the strength of those keratinocytes to stick together, would that actually phenotypically help those patients? I think that it might, which is why I was talking about, could we repurpose MEK inhibitors for other disorders in which keratinocyte adhesion is actually compromised? So this did work, which I think was pretty cool. We went to dairy disease cells and found biopsies and found that this pathway was indeed hyperactive. And these, as I mentioned, are a number of different FDA compounds that are already approved. It's been connected to adhesion already. And then I was super excited to see this case report come out that showed that trametinib was actually a promising therapy for dairy disease. This is an N of 1, but for that person, this was life-changing. They had been on all kinds of therapies. So this was super exciting to me, and I think really validates the fact that some of these molecular approaches could be used for multiple epidermal fragility disorders. The last bit is a shout out to Jess Ayers, who's also here. She's going to talk about how we think some of these similar pathways are involved in Haley-Haley disease, which is a different calcium pump that's been removed. So we use a similar model to find therapies for Haley-Haley disease. And she's actually finding that there may be connections with what Ben was talking about, the actin pathway and how it integrates with signaling pathways that, again, could be intervened upon with small molecule inhibitors. She's giving a plenary session actually later today in the Sapphire Ballroom. So I hope you'll join us for her talk as well. With that, so there are many biological and molecular therapies that are now approved for inflammatory disorders. Eczema, psoriasis they've been game changers. But we really haven't seen so many targeted therapies come to life for genetic diseases. And my lab is really aiming to change that. Using CRISPR-Cas9, we've made models of genetic blistering diseases. It can be the same thing done for PC. RNA sequencing and unbiased approach have pointed us to molecular drivers that may be common to multiple diseases, which I think is really exciting and may allow us to pool our resources. Back to the bedside, can we translate our findings into novel therapies by repurposing drugs that are already FDA approved? And could we extend this type of a platform to uncover novel therapeutic strategies for other genetic disorders? And with that, again, I'd like to thank everyone who's contributed to my research. And again, emphasize in this time of turmoil that the NIH and NIAMS have been absolutely critical to my career development and the ability for me to have this lab that does this type of research. So thank you for your advocacy and talking to your friends, neighbors, and legislators about how important the NIH is to make progress for especially rare diseases. So thank you very much.
We have time for one question.
Great talk. I was struck by the cytolysis in the basal cells in your raft model. And as I understand it, there's not an extrinsic mechanical force there. So does that mean that the loss of the keratin is driving cytolysis in an intrinsic way?
Yeah, so I think probably so. I think disrupting the ratios is probably toxic in some way to the cells. But the interesting thing is they don't just have the option to die and undergo cytolysis. Some of them make it out of the basal layer and start flattening. So I think there's ability to maybe capitalize on those endogenous mechanisms to sort of escape that really fragile state and move into a different state, maybe a more spinous layer morphology that might actually allow them to prevent the cytolysis. But yeah, they're under some amount of mechanical strain when you process them and section them and stuff. So there could be something in that realm. But yeah, it seems to be a baseline phenotype.
Okay.
Thanks so much.
One more round. So I wanted to give a shout out to two people in my laboratory that are here, Dr. Yvonne Lomakin and Min Ho. And the reason I'm calling them out now is, one, they do amazing research in my lab, and I'm very thankful for all they do. But number two, they can attest that there's probably no scientist more mentioned in my laboratory than Pierre Coulomb. Most of it is good, Pierre. So I'd like to welcome up Professor and Chair of the Department of Cell and Developmental Biology at the University of Michigan Medical School, a mentor, friend, colleague, Dr. Pierre Coulomb.
Thank you, Chris. I'm thrilled to have an opportunity to present to you about a new project in the lab. I changed my title. I'm going to tell you about a newly found link between keratin 16, which is mutated in PC, and type 1 interferon signaling. So this is currently the roadmap, so to speak, that we're using to think about PC contracting on top, contrasting what happens in healthy glabrous skin like plantar skin versus what happens in Pachyonychia congenita. And I really believe that the key defect in PC is one that consists either of abnormal sensing of cues like mechanical cues or abnormal execution of early stages of differentiation. And then you get into a faulty barrier, which leads to misregulation of immunity, innate immunity, and exaggerated stress response. And because these PC keratins are inducible and have profound non-signaling roles, you get into a vicious circle and you get into the presentation that is painful PPK. So the project that I will relate to you today is focused on K16. We've mined a number of available data sets, for instance, psoriasis, which is single cell focusing on K16 high keratinocytes, HS, which we'll hear more about today, hidradenitis suppurativa, again focusing on K6 high keratinocytes, K16. Lesional skin of PPK lesions. This is a study that was published 10 years ago by Roger Kaspar's group. Lesional skin, PPK-like lesions in our K16 null mouse model, which has beautiful PC-like PPK. That's a microarray experiment. And then finally, an interactome study where we fished for K16 interacting proteins. So we've analyze all these data sets in parallel, and basically a signature that has evolved that's shared across all these data sets, across all these analytical platforms, is one of enrichment for effectors of type 1 interferon signaling. This is true in human and in mouse. It is true in tissue and in cell culture models, and it is true at the RNA as well as the protein level. So then we reached out, by the magic of the PC project, we reached out to Alain Ovnanian in Paris at the Institut Imagine. So we knew that Alain had biopsies of PC patients. And so you can see this is non-lesional PC patient skin versus active PPK lesional skin from these patients. On this middle row here, we're staining for K16. It's prominently induced in aggregates that you can see if you're close enough to the screen for K16. And then if you look for OAS1, which is a classic reporter of type 1 interferon signaling, you see that relative to normal tissue in the PC, we have a more robust signal for OAS1. And this is also true for another biomarker of type 1 signaling shown here, ISG15, and several additional markers that I'm not showing you here. So what we see in the molecular data sets is supported by immunostaining experiments in sections from PC patients. So then we undertook to sort of investigate whether we could sort of dig further into this potential connection by using our keratin 16 null mouse model and also using a newly created keratin 6 null entered keratinocyte model. And I don't have the time to show you all that we've done. I'll show you the imiquimod paradigm in the K16 null mouse. This is activating toll-like receptors and interferon signaling. And we also did poly-IC on the keratinocytes, another way to engage toll-like receptors, a different one resulting in a type 1 interferon response. So this is the K16 null mouse model. It produces, there are thickenings at sites of contact with the ground. And if you look by histology, you have hyperkeratosis, thickened epidermis, very little lysis, if any. And over the years, we've worked the timeline. In this model, the defect starts as a differentiation defect before you see overt lesion. So if you take this mouse and you subject it to the topical imiquimod model on the back skin, which is non-phenotypic at baseline in the K16 null mouse. You compare the response of wild-type mice or K16 null mice to the imiquimod paradigm. You see that at an earlier stage, and then by day 5, a much more profound reaction in the K16 null. The tissue is crusty compared to wild type, and if you do histology, there is an exaggeration in the thickening, in the proliferation, several other index of stress response in the skin. Of course, K16 is induced in this paradigm, but is missing in the K16 null setting. So if you look for reporters of type 1 interferon signaling in this model, you see that in the wild type, as is known for type 1 interferon responses, it is mostly concentrated in the basal compartment of the epidermis. You lose that polarity in the K16 null in that if you quantify the images and several markers, you see that as opposed to have the phasing down of the signal as you move up in the epidermis, it stays very, very high in the suprabasal layers where K16 should be, but is not. Now, just moving to the cell culture model, we're working with de-entered keratinocytes. We found a way by culturing them to post-confluence and then waiting patiently that they form a bilayer. The bottom layer is K14 positive. The bottom layer is K10 negative, K16 negative, filaggrin negative. But the suprabasal layer is K10 positive, K16 positive, and filaggrin positive. Finally, we have a model where K16 is expressed, where it should be expressed in human keratinocytes ex vivo. So then if you take this model system, and I apologize for the complexity of this slide, this is the roadmap that we used on the left. We treated these cells with poly-IC, and you can see that relative to wild type, the knockout has a greater amount of ISG15 expression and a greater amount of phospho-IRF7, which is one of the transcriptional regulators that's engaged in response to the stimulation of the pathway. The data is quantitated in the bottom. And if you look at the cells themselves, this is here ISG15, this is your wild type control, and this is the K16 null. It's high, and this is phospho-IRF7 in the nucleus, which is also higher. So how is the connection between K16 and interferon signaling regulation made? We believe that a key component is 1433 epsilon. 1433 epsilon is a keratin 16 binding protein. We have that by co-IP. We have that by proximity ligation assay. And if you look at the interaction between 1433 epsilon and RIG1, which is an early effector of type 1 interferon signaling responses. And so here we're looking at the cells, and here we're looking at the tissue from the mouse, the imiquimod model. You see that the interactions between these two proteins, which indicates type 1 interferon signaling, is dramatically enhanced in the absence of K16 relative to control. So the conclusions of this body of work, which is currently under revision, is that induction of K16 in differentiating keratinocytes spatially restricts the type 1 interferon response to the basal layer keratinocytes in stressed epidermis. Type 1 interferon signaling has been implicated by others in the development and homeostasis of villus skin, and Luis Garza at Hopkins has shown that for the hair follicle regeneration paradigm, interferon signaling is very important. So we believe that there is a heightened activity of type 1 interferon signaling in PC skin, and the consequences for PC pathophysiology at this point are unclear. We're working to investigate that. Whether this would offer a new therapeutic opportunity is also unclear. But one thing that's interesting to remember is that given the prominent role of interferon signaling in antiviral responses, one might want to think about this when trying to improve siRNA-based strategy to treat dominant diseases like PC. So with this, I thank you for your attention. I want to thank the people who have been driving this project over the years, the people in the lab, the collaborators. Eris Cohen is a postdoc who is the main protagonist here. Amanda Orozco is here. She's a contributor as well, and she has a poster on the new mouse model for PC a bit later on in the week. So thank you very much.
We have time for one question. To the right.
Thank you very much, Pierre, for this wonderful talk. I was just wondering whether you may perhaps also
У нас
think of
книжечки есть
applying JAK
дома.
inhibitors in order to rescue your
The
phenotype, and
snake
that
on
may
the way.
have some translational implications as well.
Мы
Which
еще
JAK
смотрели, да?
inhibitors?
Режим ДВ. Какие ты хочешь? Супер, у нас как минимум мультик мы смотрели. Можем еще посмотреть дома мультфильм. Надо книжку заказать.
I have the name in my computer. I can tell you.
Well,
But one has to be very, I'm aware that depending on, there's many of these products that are available, and we've been.
Well, I'll give you my thoughts.
Yeah.
That may be the only one approved, but what I would tell you is that we just published the type 1 interferon signaling paper on the structural mechanisms of TYK2 inhibitors. And so it turns out that TYK2 inhibitors, particularly the next generation zasocitinib, is a very potent type 1 interferon signaling inhibitor. So what I would like to do is try zasocitinib in PC patients with K16 mutations. I think that is our best bet.
Yeah.
Okay, so it is a rare opportunity that you get to introduce your own chairman to come up and speak. And so Dr. Keith Choate has literally, I think, taken a red eye after cancellations to get here to speak to us today. Keith is chair, professor and chair at Yale, and certainly a mentor to me. And without further ado, Keith.
Thank you. Thank you so much, Chris. So one of the things that I've loved about being a part of PC Project is how deeply embedded it is in thinking about patients and thinking about genetics. And so I wanted to tell you a new story that's come out of the lab, but really also to point you to my students who will be presenting this at the concurrent session and also at the poster session. In terms of disclosures, I'm an investigator, scientific advisor, and a consultant. So I approached genetic skin disease from the world of a different sort of epidermal differentiation disorder, primarily thinking about ichthyoses, but we recognize that ichthyoses also have the feature of palmoplantar keratoderma. And as we are recruiting patients from time to time, we sweep in PPK patients. We always send the PC patients to the PC project, but others get carried through. And so in the course of creating a registry over the last 15 years, we recruited about 1,800 kindreds. We've been able to provide genetic diagnoses to about 1,450. And we've become much better, partly through observing the work of the PC project at phenotyping our patients and also screening them effectively for mutations in 72 genes. And those patients who don't have mutations in known genes go on to genome, exome, or transcriptome sequencing. And they've also served as an incredible resource for us to do other work in terms of generating severity scores and understanding more about disease pathophysiology. And so we take a genetics forward approach. That is, those patients who don't have mutations in known genes go through exome and genome sequencing, and then we look for recurrent mutations. And we found a very odd thing. And that was we had multiple individuals who had dominant de novo mutations in SLRP1. Now, SLRP1 is known to cause a recessive disease, malda melita. So we instantly went and looked at the photographs of these patients and said to ourselves, well, they sure don't have Maldomlita, because while one has a very severe form of PPK, the other has progressive symmetric erythrokeratoderma, which has not been seen due to mutations in SLRP1. And we have nice dominant transmission through multiple generations in this other PSEK kindred. So this suggested to us that, in fact, this might be a new disorder with a distinct mechanism of action. And so we began the process of trying to figure this out. So we started by thinking about SLRP1 itself. We know that SLRP1 is a secreted protein. It has a 22 amino acid signal peptide sequence, which is cleaved during secretion. That's going to be important because these mutations actually affect that signal sequence. We know that SLRP1 acts on alpha-7 nicotinic acetylcholine receptors as an allosteric agonist and also interacts with the receptor outside of the classical ligand binding site. The more I read about this, the more I came to understand how little we actually know about the function of SLRP1. It has a three-finger structure with beta structural core through prolonged loops. It's expressed in the granular layer of the epidermis, induced its differentiation, and it's been shown to inhibit keratinocyte proliferation in vitro. The majority of cases of Maldomlita involve truncation mutations that affect different portions of loop 1 and 2 and the head domain. And these primarily lead to loss of function of the encoded protein. So I wanted to take a phenotype first approach. So shown here is the phenotype of one of our PPK cases. And you can see this is an extraordinarily severe isolated PPK. However, we also had patients that had a different mutation. This was an H22D mutation, and you can see that they have a milder PPK, but they also have psoriasiform plaques, which are quite extensive across the body, as you can see in our next kindred here, where we have these widespread psoriasiform plaques and a smoother palmoplantar keratoderma. So the first thing that we did is we looked to see what the consequence of these mutations could be. So both of the mutations occurred at the terminal residue of the signal peptide sequence of SLRP1. We used signal P to look at what these mutations might do, and what we predicted was that, in fact, what it might do is cause shifting of the cleavage site to amino acids upstream. So we overexpressed wild-type SLRP1 in each of our mutants, and then looked at the secreted protein, and in fact, this is the wild-type sequence, LKC. We find that H22D adds ED and H22V adds EB to the produced protein. So we were clearly getting a novel peptide, which might have a neomorphic effect. So we then went to cells from one of our patients who had the PPK phenotype and one of our patients that had the PSEK phenotype and said to ourselves, well, are they making SLRP1 at all? And what we found is that, in fact, they were actually hyper-secreting SLRP1. So we took healthy keratinocytes and keratinocytes expressing PPK and PSEK variants, basal state differentiated them, induced expression of SLRP1, found we had a massive overproduction of SLRP1 in our PPK and PSEK patients when controlling for medium actin. We also had increased cell SLRP1. This correlated with immunofluorescence staining, where we see really relatively mild staining of SLRP1 in healthy control skin, but quite expanded and exaggerated staining in both PPK and PSEK cases. So this was really fascinating to us. And so we realized that we needed to get more granular understanding of what was going on inside of the tissue of our patients. And so we took on this by performing high-resolution spatial transcriptomics. And what we found was that, in fact, spatial transcriptomics very quickly showed us that, in fact, SLRP1 was increased in expression in terms of the transcripts, but it also showed us that we were getting early induction of skin terminal differentiation, which relates back to an earlier talk that we heard, but also strong innate immune activation, which relates to another talk that we heard. We also found that NF-kappa B signaling was identified as one of the top enriched pathways in these patients. So we also remembered that there were these psoriasiform plaques. So we wanted to understand how similar these plaques were to true psoriasis. And so we did RNA in situ hybridization to look at expression of IL-4, 13, IL-17, and IL-36. And what we found was that, in fact, there wasn't increased expression of IL-17 in our PSEK patients, in contrast to psoriasis patients, but that we had a significant induction of IL-36 in our PSEK patients, similar to what we see in psoriasis, and something that we're seeing universally across ichthyoses more generally. And we were extraordinarily lucky to have wonderful structural biologists who I may call upon to speak more about their findings. But we asked this question because we were seeing a profoundly different phenotype on the basis of a single amino acid substitution. We said to Dr. Bunick and Dr. Lemakin, how is this happening? And so they said that the binding surface of SLRP1 involves the loops of SLRP1, and the head and end terminus are positioned on opposite sides of the receptor binding interface, indicating an allosteric binding. And so they suspected that, in fact, these A22 variants may be differentially affecting binding of a previously hypothesized secondary target that binds to SLRP1, and that, of course, it can also affect cleavage and secretion kinetics that may partly explain the distinct phenotypes for this. And I may need them to step in to tell us more. So altogether, this was, I think, a classical example of how we can go from using unbiased genetic approaches, find patients that clearly haven't read the textbook, and identify a new genetic cause of disease, but also achieve meaningful phenotypic expansion, which tells us something about the biology of PPKs in general. Here we find that variants at the A22 position can cause either severe PPK or PSEK. They do lead to an alternative signal peptide cleavage site, which induces differentiation-induced expression and secretion of SLRP1. The question of how that different secreted protein is actually exhibiting its effect to lead to NF-kappa B activation remains one of the unanswered questions. But I think that these give us potential new targets for thinking about treating at least this PPK, focused on NF-kappa B and increased innate immune activity. And I, like many others, will be thanking the NIH, which funded my lab, and Dr. Bunick's, the Pediatric Dermatology Research Alliance first, and we have a deep collaboration with PC Project as well, and to our patients who made this all possible.
We have time. We have time for several questions. Ellie.
Thank you very much. I thank you very much, Kate, for this wonderful talk. First of all, maybe a small remark. When you look at patients with maldomeleda, they have usually much more than palmoplantar keratoderma. They have a lot of extra palmoplantar involvement, maybe not extensive as the one that you show in one of the two cases that you show, but still they can sometimes remind you of psoriasis. They have a lot of perioral or periorificial involvement. So surely the mechanism is different. I completely agree, but still, from a phenotypic point of view, it can be sometimes difficult to distinguish even those from, we've had some patients refer to us with psoriasis, actually.
And I think you're actually onto something here, Ellie, because my understanding is that no one has actually looked deeply enough at whether we actually have expression of SLRP1 in the majority of those psoriasiform cases. And so I think it's likely pointing to something mechanistic.
Okay. So my question was, do you have like direct evidence for this slightly modified SLRP1 as that protein, that modified protein, which is inducing early differentiation, like, for example, introducing the mutant into keratinocytes or doing some, or is it just correlative at this stage?
So at this particular moment, it's primarily correlative. We have cell lines that are now expressing this. We have skin equivalents that are expressing it. We do see that we can reproduce the phenotype by overexpressing the mutant, but that's what we would expect. I think that there's a lot more work to do.
Thank you.
Hi. My question is regarding the interleukins that you have seen, and especially IL-36 gamma. If you looked also at the blood levels or the secreted levels of these interleukins in the patients. And follow-up question, if you try to see if there is also effect on IL-8?
So I think we are aware that there have been clinical trials of IL-36 inhibition in certain types of ichthyosis, and it's my understanding that some of them have been terminated. The question is whether IL-36 is simply an alarmin that's expressed as soon as you get a stress response within cells. I think that that's likely the case, and so IL-36 may be somewhat non-specific. As to the question about IL-8, I don't have a good answer for you today.
Along those lines, do you think spesolimab would work in these patients?
I suspect not.
It's trickier than what I think.
So we've actually used IL-36 inhibition in other ichthyoses in mouse models. And despite the fact that we can effectively reduce IL-36, we see actually a worsening of phenotype rather than an improvement of phenotype. That's unpublished data.
Okay, it is 10 o'clock. So we are going to do a group photo. So I invite our fearless leader to come tell us how we're going to execute this.
Hey, thanks, you guys. This is really great. Just a quick announcement for all of you on Zoom. We're going to take a 15-minute break. And so just know we love you. And thanks for joining us. We care about you. I want to give a special shout out to Dave Hansen and Adele O'Toole, who've been watching from afar, so their hearts are with us. With that, don't hate me. We're going to do it. You know, we have to get the picture. We have to prove how awesome we are. And so what we're going to do, you guys are, a lot of you are doctors, you go to fast appointments, just pretend there's a patient on the table and go that way as fast as you can because they've been waiting for 45 minutes. Just kidding, but go, Michael, do you want to wave? Let everybody look at Michael. And he is going to lead you. We're just going to go straight that way because he scouted out a nice view for us. So if you are in this room, and it's nice to see Dr. Dyer here. I haven't met you yet, but I've been wanting to, so I'm thrilled you joined. Everybody, you're part of us, so come to the picture, and then you can have your break. Pro tip, we're cheap. We didn't buy you a snack. We just bought you drinks. So your snacks, don't tell Hilton, but your snacks are all on the table right here. So come find a seat so you can have some food. Okay. With that, follow Michael quick.
Can you hear me? Are you hearing me okay? Yeah, are you hearing me okay? Excellent.
If everybody would grab a seat, we could get started on the next session. Chris did such a good job chairing the last session. He ended exactly on time, so I'm going to try to do the same. We have our first speaker in this session is going to be Michael Caterina from Johns Hopkins School of Medicine. He is focused on the cellular and molecular mechanism of pathological pain, and he's going to be speaking today on this subject specifically for PPK, and really excited to hear what he has to say. He's going to join by Zoom, so.
Thank you very much. Can you guys hear me okay? Well, are you hearing me now? Great, thank you. So what a pleasure it is to be able to join you today. I want to thank Janice, Holly, Pierre, and all the organizers for allowing me to present virtually today. I'm sorry I couldn't be there in person. Also, congratulations to Janice and her family for little Benny. That's great news. I was introduced to the world of PC and PPKs about 10 years ago by Pierre Coulombe, who I think, as we can tell from today's comments, is a little bit like the Pied Piper of the field, sort of bringing all these investigators from various fields in to study these very challenging medical problems. And in my case, became very interested in this because my laboratory is interested in understanding how we sense pain. What are the events that lead us to sense augmented pain when there are conditions going on in the body that lead to abnormal pain sensation? And so what our inroads into this problem were to see whether or not we might be able to utilize mouse genetic models of palmoplantar keratodermas to try to understand something about why some forms of PPK are more likely to be associated with pain than others. So in general, Pachyonychia Congenita and Olmsted syndrome are almost universally associated with intense pain or itch, whereas in other conditions like multimilata, epidermolytic PPK, pain can occur in some patients, but it's not quite as widespread. And yet, if you look at the histology of all these conditions, at least to a non-dermatologist like myself, the skin histology looks fundamentally similar in many of these conditions. So we'd like to understand at a cellular and molecular level what's going on here. And at the end of the talk, I'll give you a little bit of a sense of where we've gotten with this. So the mouse models that we use the most in our laboratory are mouse models of a condition that we heard about in the last talk, and that's multimilata. Steve Young at UCLA had generated both SLRP1 knockout mice and SLRP2 knockout mice a while back and shown that these mice exhibit palmoplantar keratoderma. They exhibit pronounced thickening both in the hind paws and the front paws. And we got a really nice introduction to SLRP in the last talk, so I won't go into detail regarding what it's doing molecularly. except to say that we're taking advantage of this pathology to see if we can understand something about pain. And our question when we got a hold of these animals was whether or not they exhibit any sensory abnormalities that we could document. And so these are the results. This is work from Rachel Weinberg and Dennis Chang and Suyun Kim in my lab. And what they were able to show is that both SLRP1 knockout and SLRP2 knockouts, which basically phenocopy one another, exhibit increased mechanical pain sensitivity. And this is quantified here by plotting how many times they withdraw their paws when you administer a certain force to the hind paw. And you can see that in both cases, these blue bars are bigger than the black bars, indicating that these animals respond more to mechanical force. They are also more responsive to painful heat stimuli, and that's shown on the left-hand side here, where if we shine a heat lamp onto the bottom of the foot of one of these mice, and we measure how long it takes for them to pull their paw away, we see that it takes the SLRP knockout mice less time to pull their paw away than it does their wild-type littermates. And that's true for both SLRP1 knockouts and SLRP2 knockouts. Now, importantly, if we heat the tail skin of these animals by dipping in a hot water bath, we don't see any difference between wild-type and knockout animals, showing that this increased sensitivity really is confined to the skin tissue that's affected by the PPK. We also see that there's increased pain-related behavior in these mice when you don't do anything to them. Just with them walking around on a surface, they show increased paw flinching, paw licking, and other signs of spontaneous or ongoing pain sensitivity. If we look histologically at the skin, what we find is that there are dramatic changes in multiple immune cell populations in the skin of these animals. So, for example, there's an increase in T cells labeled by CD3. There's an increase in mast cells labeled by avidin. There's actually a reduction in epidermal Langerhans cells, and there's an increase in macrophages. And so we're very interested in what they may be contributing to the pain phenotype. In fact, before I won't show you the data for this, but this prompted us to test whether an anti-inflammatory agent, an aspirin-like substance, could attenuate pain sensitivity in these animals. And indeed, we can get a partial reversal of their pain hypersensitivity with these compounds. Lin Tao Chu, who was in my group, showed physiologically that if you record directly from the sensory neurons of these animals under deep anesthesia, while you're poking at the hind paw, you get an increased not only in their responses to mechanical stimulation, but in some of these neurons, you have electrical firing even without any stimulation, showing that there is a fundamental physiological hypersensitivity in these animals. Now, that has prompted us more recently to take a look to see if we can identify gene expression alterations that might be able to help explain the pain hypersensitivity in these SLRP knockouts. And so we have started this out by doing bulk RNA sequencing, both on pad skin and on the dorsal root ganglia. These are these collections of neuronal cell bodies that provide nerve terminals to the skin. And we saw, I won't go into detail here, but we saw something like 5,000 gene expression changes in the skin of these animals. And we've been working together with Pierre Coulombe and Craig Johnson and his group to start to compare those gene expression changes in the skin with what's seen, for example, in the keratin 16 knockout model of PC. But where we've been focusing most of our attention is in the sensory neurons. In the dorsal root ganglia, we see a much more manageable list of about 40 genes that seem to be dysregulated based on bulk RNA-seq analysis. When we went to confirm this by qPCR, when we went to confirm this by qPCR, we see that a number of the genes that show up in this analysis, we can confirm as being upregulated in the dorsal root ganglia of both the SLRP1 knockout mice, and I'm not showing you here the SLRP2 knockout mice. A couple of important points about this. One is that right for the time being, we're focusing on these 4 genes right here. PAPA, which is a protease that I'll tell you more about. TRPA1, which is an ion channel, which we know to be involved in pain and itch sensation. BDNF is a neurotrophic factor that people have known to be involved in pain. And this other non-acronymic molecule, VGF, is another injury-associated trophic factor that's been associated with nerve injury-induced pain. The other thing I want to point out here is that these genes are not upregulated in sensory ganglia that are innervating the trunk skin of these animals. So this upregulation, again, seems to be very intimately associated with the sensory neurons that innervate affected skin, which suggests that there may be a cause and effect relationship here. And then on the right-hand side, we confirmed by using in situ hybridization, a method called RNA-scope, that increase in mRNA, for example, for this gene PAPA, is confined to the sensory neuron cell bodies rather than the many other cell types that you find in sensory ganglia. Now, another reason that we've thought that this is going to be worth pursuing further is because we've taken advantage of other mouse PPK lines, for example, the keratin 16 knockout line that Pierre Coulombe's lab had generated, and the keratin 9 knockout line generated by Erwin McLean's lab. And both of those lines exhibit PPK, particularly in the front hind paw, or in the front paw skin, on the pad skin. And when we did pain testing on these animals, what we observed is that whereas there is pain hypersensitivity in the keratin 16 knockout mice, there is no such hypersensitivity in the keratin 9 knockout mice. And we can see this in this heat assay, but we also see it when we look at their spontaneous pain behavior, which isn't shown here. So that prompted us to take a look at the ganglia that innervate the front paws in these two mouse lines. And the data are shown in somewhat condensed fashion here, but I want to point out is that, these same 4 genes that are upregulated in the lumbar dorsal root ganglia that innervate the hind paws of the SLRP knockouts are also substantially upregulated in the ganglia that innervate the front paws of the keratin 16 knockout mice, and not change at all in the ganglia that innervate the front paws of the K9 knockout mice, which don't show a pain phenotype. So in other words, we have a very nice correlation now between this handful of genes and their upregulation in the sensory neurons that innervates PPK skin when there's abnormal pain sensitivity, but not when there's not abnormal pain sensitivity. Now, coming back to this gene right here, PAPA, it encodes a protease that's secreted by cells, binds to the extracellular matrix, and the role of this protease is to cleave IGF binding proteins. There's a series of these proteins, and what they do, these green structures, is that they normally bind insulin-like growth factors 1 and 2. And when IGF-1 or IGF-2 are bound to these IGF binding proteins, they can't access their receptors. So as long as they're bound, they're biologically inert. However, when PAPA cleaves one of these IGF binding proteins, that liberates the bioactive IGF, and it can now activate its downstream receptor. Now, we don't know yet whether there is an increase in IGF-1 receptor signaling in the SLRP knockouts. That's work that's currently being conducted. But what Soo-Yeon Kim in the lab has been able to do in collaboration with Cheryl Conover and Lori Bale at Mayo is to show that she can actually measure the activity of this protease called PAPA in the dorsal root ganglia of our mice. So this is just a biochemical assay showing that an IGF binding protein can be cleaved by membrane preparations from these sensory ganglia. And that cleavage can be blocked by adding a specific antibody that neutralizes the activity of PAPA. We also have gone on now to cross the SLRP knockout mice with PAPA knockout mice that we got from Cheryl Conover's group. These mice are actually smaller than their litter mates because of the role of PAPA in insulin-like growth factor signaling. What you can see is in the double knockouts, we don't see any change in the PPK. The skin is still thickened in these animals. But what our preliminary data suggest is that the mechanical hypersensitivity, the pain augmentation that we saw in the SLRP knockout mice, is partially attenuated when you concurrently knock out this PAPA gene. So to summarize these data, we found that SLRP1 and SLRP2 knockout mouse models of multimilata can exhibit a multimodal pain hypersensitivity, spontaneous pain behavior, neuron hyperexcitability, and substantial alterations in skin immune cells. They also exhibit alterations in the expression of thousands of genes in paw skin, but a smaller number in lumbar dorsal ganglion neurons. Many of these mRNAs are upregulated in the limb innervating dorsal ganglion neurons, but not in those that innervate other parts of the body. Some of these mRNAs are also upregulated in other painful PPK models, but not in those models with normal pain sensation. So we think that they may be good handles to go after as potential contributors to the pain phenotype. And finally, PAPA might represent an important contributor to the pain hypersensitivity, not only in the slurp knockout setting, but perhaps in other palmoplantar keratodermas that feature pain. Finally, in terms of future directions, there are really two big directions we're going with this. One is to go gene by gene now with some of those genes that we've identified so far to see if by knocking them out or by blocking them pharmacologically or neutralizing them with antibodies, we can interfere with that excessive pain phenotype that we see in the slurp knockout and keratin 16 knockout mice. The other approach that we're taking is something we're doing now with the generous support of the PC project, and that is to take a single-cell RNA-seq approach. Sorry, this should be single-cell RNA-seq across PPK models. And the motivation for that is if you look at this slide of a dorsal root ganglion, you see that there are many different subtypes of sensory neurons in our sensory ganglion, at least 15 different subtypes. And by looking by single-cell RNA-seq, we can start to cluster them into different subcategories that have functionally distinct importance. And so what we've seen with other projects in the lab is that there are genes whose differential expression will show up at the individual subtype level that we never would have seen at the bulk-seq level. And so with the help of this PC project grant, we are comparing slurp knockout mice, keratin 16 knockout mice, and the keratin 9 knockout mice with respect to gene expression differences at the single neuron subtype level, hopefully to identify additional targets that we might be able to validate and go after therapeutically. So with that, I just want to acknowledge the people involved in this lab. Rachel Weinberg, a former graduate student in the lab, and currently Soo-Young Kim and Tripti Tripathi are three graduate students who have been heavily involved in this work. Dennis Chang is a postdoc in the lab who's contributed this work as well. Lintao Chu is a former assistant professor who had worked on this project. And of course, we've enjoyed a years-long enjoyable collaboration with Pierre Coulombe and his colleague Craig Johnson, who are helping us with the single-cell PCR work. We could not have done this work without the support of the Neurosurgery Pain Research Institute at Johns Hopkins. As I mentioned, we're very fortunate to have some support from the PC project now to engage in this single-cell RNA work and other projects in the lab. I'll just echo what others have said. We wouldn't be a lab without NIH support, and these are just a couple of examples there. So with that, I thank you all for your attention and for allowing me to present remotely.
I think we have time for one quick question. We are running a bit over, so if we don't have questions, it's thank you. I don't know if you can hear me, Michael, but thank you for that great talk. I couldn't even pay attention to what you were saying for the last slide because that image was so unbelievable. It's a really beautiful work. Thank you.
Thank you very much.
Our next speaker is Diana Bautista. She is a Howard Hughes investigator. Do you want to stand on? You want to stand on this side? Or you want to stand with the mic? Okay. I don't even know. Let me find the slide changer. Okay. All right. We're here. If you want to stand over here, I'll hand the mic over. Diana Bautista is a Howard Hughes investigator at UC Berkeley. Today, she's going to speak to us about neuronal hyperexcitability, pain, itch, and inflammation. You can take the mic or you can use this one.
So how's this? Okay, great. Well, thank you so much for the opportunity to be here. I'm excited to tell you about some of our work looking at atopic dermatitis associated itch and pain. So just generally, my lab is interested in neural mechanisms underlying itch and pain under healthy as well as disease conditions. We spent over a decade looking at sensory receptors, ion channels, and signaling pathways that respond to a variety of inflammatory mediators, including proteases, serotonin, inflammatory lipids, and inflammatory cytokines, to show that these signaling pathways are key for triggering itch and or pain, again, under healthy conditions, but also in a variety of disease models. And about 10 years ago, became very interested in understanding neuroimmune mechanisms underlying atopic dermatitis, one of the most common chronic itch disorders that's characterized by these red, dry lesions that are found in both children and also the adult form of atopic dermatitis. And also, of course, a key hallmark is really severe, debilitating itch that's difficult to treat. Antihistamines are completely ineffective. We became interested in this because we were interested in neuroimmune interactions, but also atopic dermatitis is an entryway disease to other types of atopic diseases. So even though in many individuals, as you age, the atopic dermatitis could improve, we know that then there's a following of a food allergy, allergic rhinitis, and also asthma and what's known as this atopic march. And again, these sites are also densely innervated by sensory neurons that we know can interact with epithelial cells in the airways, as well as the skin, and also influence the immune response. So we became interested in probing this in a model. And also we know, and this has been really highlighted in recent clinical studies, that at least 50% of atopic individuals report severe pain, that this pain is not correlated with itch or scratching or even the lesion itch scratch cycle. And the descriptors are associated with neuropathic pain descriptions, and that it also contributes, like itch, to decreased quality of life. And the mechanisms are largely unknown. So we thought this is a great opportunity to look at both itch and pain mechanisms. So Takeshi Morita in my lab adapted the MC903 model of the atopic march, where topical application for 14 to 60 days triggers skin barrier disruption, immune cell infiltration, and eventually the atopic march to allergic airway inflammation. We adapted this to the cheek model. Typically, it was done in the ear or the neck. And this allows us to distinguish between itch and pain behaviors clearly, where mice who are itchy scratch with their hind limb, and mice that are in pain will rub with their forelimb. And we adapted this to only a 7-day treatment because we wanted to look at the development of atopic lesions and itch during the acute treatment, but also wanted a chronic model where we could look at the mechanisms that change in the absence that stabilize itch and pain in the absence of doing anything to the skin. And indeed, the 7-day treatment triggers robust lesions that start around day 3 before the onset of itch, and they persist long past the treatment period. And we also observed robust scratching behaviors in this model that begin around day 5, and they increase, as quantified here, during the treatment period, but they persist several weeks post-treatment. And this gives us a nice opportunity to look at the mechanisms that drive the early acute itch, as well as that transition to a chronic state. And we also, recent data in the lab has shown that beyond that treatment period, starting around day 9 and really showing robust behaviors around day 12, is that these animals also display wiping behavior and this really unique head shaking phenotype where they wipe that local treated area and then shake their head following that we don't see with other things like capsaicin. So we're studying this behavior and the mechanisms as well. And that's just quantified here on day 12. See a lot of wiping. So because atopic disease span tissues and time, we took a comprehensive systems physiology approach to understand this. So Rose Hill and Carolyn Walsh in the lab looked at atopic dermatitis behaviors. They dissected many tissues from skin to the sensory ganglia to the lung, and we've recently added spinal cord and brain. And we're doing things like RNA-seq, sorting immune cells. looking at histology, and we've been doing mass spec to look at lipidomics as well. And I'll just show you a small subset of these data. We think this is a really good model. Just looking at the skin across the first 8 days of this model, we found that 70% of known human atopic dermatitis associated genes were dysregulated. And now we have a nice temporal window of when these different genes are being up or down regulated. And looking across multiple types of itch disorders, we found that 62% of known itch genes were dysregulated as well. And we're following up on what these different genes are doing and when they're doing them during this model. Remarkably, after just a single treatment and within 6 hours, we saw really dramatic skin barrier dysfunction and dramatic upregulation of proteases and decreases in key skin barrier disease genes. We think this really sets the stage for everything that follows. We also saw by day 2 really dramatic hyperinnervation of the skin that was transient. It lasted for about 24 hours. And characterization of this innervation showed that all of this hyperinnervation were the TRPA1 positive population of neurons that Michael just mentioned that are able to promote the release of inflammatory neuropeptides that can recruit immune cells, drive the immune response, and also trigger hypersensitivity of sensory neurons. Following this, we saw a dramatic increase in a variety of key cytokines and chemokines in the skin, including TSLP and a variety of other chemokines. And this was followed by the early changes in recruitment of neutrophils to the skin. And it wasn't until day 8, after itch was in full bloom, where we saw the classic type 2 immune signature of T cells and basophils and these inflammatory cytokines. So we wanted to ask, what's happening early on? Are these TRPA1 positive sensory neurons required for the development of itch and AD pathogenesis? And what are those early neutrophils doing? When we looked at TRPA1 knockouts, or we used pharmacological agents, we found that itch-evoked scratching, both in the early phase and the persistent phase, was significantly attenuated. And interestingly, we found that lesion formation actually that precedes scratching, so it's not a skin damage response. These lesions depended on a functional sensory neuron and TRPA1 ion channels, suggesting that these sensory neurons are not just passive players transmitting itch, but they could actually contribute to early disease phenotypes. And we also found that there was this key interaction between these TRPA1 sensory neurons and neutrophils that depended on CXCL10. And so CXCL10, if we block the receptor, its receptor CXCR3, or we deplete neutrophils, again, we saw a significant attenuation of itch, both during the early acute phase and during the persistent phase, up to a week and a half past the last treatment of the skin. So we think that there's different sets of neuroimmune interactions. Early on, there's this neutrophil sensory neuron interaction that depends on CXCR3. And then later in the chronic phase, we and many others have shown now that there are different subsets of sensory neurons that express a variety of key type 2 cytokine receptors that interact with basophils and T cells to drive a more chronic itch that has a more intense itch severity. So what mechanisms promote pain and airway inflammation in this atopic march? This is a big topic. We started to address this, like Michael Katerina just showed us, by looking at the dorsal ganglia, looking at gene expression changes. We saw that there were about 70 genes that change expression. They were related to changes in excitability, immune and neuroinflammation, as well as immediate early genes that we know are key hallmarks of neuropathic pain. So we're investigating this as a mechanism for driving the pain associated in the chronic phase. We also see a really dramatic increase in the number of immune cells in the sensory ganglia that persist several weeks after the treatment period. And these include CD8 T cells, B cells, and macrophages. We also see dramatic changes in the dorsal spinal cord starting around day 8 of this model. right about where we start to see pain behaviors. And this includes hallmarks of demyelination, differentiation of the oligodendrocytes seem to be defective, and we also see some hallmarks of demyelination in the trigeminal ganglia as well. And we also see clear signs of reactive astrogliosis in the dorsal spinal cord as well, again, indicative of not only really severe chronic itch, but also neuropathic pain. So we're trying to figure out if this is driving itch, pain, or both in the model. And we have started to look at the brain. And again, starting on day 8 of this model, we see an increase in B cells and basophils in the brain. And by day 12, we see really significant increases in B cells, as well as B cell cytokine production. And we know that B cells are linked to brain inflammation and a variety of disorders, and that these B cell cytokines are a key player in neuropathic pain mechanisms. So we think that this early changes in neuroimmune interactions and inflammation in the skin drives this temporal wave of inflammation that spans from the periphery to the central nervous system. And we're very interested in trying to decipher these neuroimmune mechanisms and exactly what they're doing in this model. And we're also defining the neuroimmune interactions, the atopic march, and airway inflammation. And in parallel, we see dramatic changes, almost sort of a phenocopying of the neuroimmune epithelial cell interactions that we see in the skin 8 days later that show up in the airways. And again, probing a key role for TRPA1 sensory neurons in this process. So with that, I'll just thank the people who did the work, my funding agencies, and thank you for your time today.
We have a couple of minutes for questions if anybody has any.
Maybe I'll ask a wonderful talk, a really basic question. Clinically, for both atopic dermatitis and some of the other skin disease patients, that they oftentimes are describing burning sensation. Is that more of a pain related, or is it a more itch?
Yeah, it's a great question. And it's hard to know. We can't tell in our mouse model. We're happy we could distinguish between itch and pain at all. But I think that, with, scoring itch and looking at questionnaires from, clinical questionnaires, there's hundreds of descriptors for both. And there's a lot of overlapping descriptors. And I think that makes it so tricky in knowing what to do. There's also this really dramatic interaction between itch and pain, where pain inhibits itch, scratching causes pain. And I think this creates a big confound to try to answer that question as well. And also, I would say that touch sensitivity dramatically changes as well, such that light touch now becomes irritable and, and itchy. So all of the sensories seem to be shifting in different ways. Those, are the sensory experience, making it quite, I think, challenging.
Diana, that was lovely. You mentioned that the process continues sort of after the treatment, but then you keep looking. What about how things start to resolve, and what can one learn from that in terms of, let's say, itch or pain going away?
Yeah, it's a great question. We did not think it was going to persist for so long. So we thought we're going to induce, we'll see a chronic phase, and then we could look at the mechanisms that allow it to resolve. And there's just been so much to do looking at these the first 2 weeks. That we haven't gotten to the resolution side. And I think it's going to be really important because we know that these atopic lesions are really cyclic. There's bouts where you have these lesions, where you have itch, periods of remission, and what's the trigger. I should just along those lines, though, these neutrophils that we see in the few clinical cases, studies that are out there where either a biopsy was done on a forming lesion, on a pre-lesion, or where lesions were induced actually in the clinic, neutrophils are there clinically. So we don't think it's just a weird mouse thing. And that, I think, there's a lot of drugs out there for neutrophils for other diseases, and maybe we could convince people to take a look at that. I have atopic dermatitis, so I know when my lesion is coming. And so I'd love to be able to put something on it to stop it from forming.
Cool, thank you. We know that pain is the certainly the worst symptom in PC, but I think that deep itch is probably certainly second. And so it's really nice work. Thank you for that. The next speaker is Amanda Nelson. She is an associate professor at Penn State, associate professor in dermatology at Penn State, and she is going to be speaking to us today about her group's approach to identifying and developing therapeutics for HS.
Great. Thank you so much. Thanks to Janice and Pierre for welcoming me to give a talk on something completely different. So my lab studies HS. And so I do have some disclosures. Insight funded a clinical trial that I'll talk a little bit about, and I also consult for them on other projects. Okay, so this is first and foremost why we study HS. It's the patients. So this is Athena. She's one of our Penn State patients. I've gotten to know her really well over the last 15 years. She's an HSF, hidradenitis suppurativa board member, and she also says she is living with HS. That's her quote. She lives with this disease every single day. And these are some of the questions that she's asked me over the last decade or so. What exactly is this disease? Why do I have this? And to her, what can I do when I have a mild lesion coming on? Okay, so just so we can all get on the same page, this is HS. These are what the lesions look like. Okay, it's a chronic, relapsing, remitting inflammatory condition that favors the skin folds. So this is your armpits and your groin. For women, it can be under the breast. Sometimes in the male, it can be around the waistband or even in the neck folds. It has very deep-seated nodules. So these are nodules that go deep down into the dermis, almost to the level of the subcutaneous fat. There's abscesses, sinus tracts, and eventual scarring. Pain, itch, and drainage and odor are the chief complaints from these patients. The prevalence is about 1% in the US. Some literature says it could be up to 4%, but it is definitely underdiagnosed because patients don't seek medical care. It's 2 to 3 times more frequent in Black people, and we really don't understand why, and it's 3 times more frequent in women. The onset is sometimes between 10 or your tweens and 20s, but it can actually be later as well. But it's not just the skin lesions. This has a severe negative quality of life for the patients. It disrupts their work, they lose jobs, they lose promotion opportunities. They have high medical bills. On average, the patients spend roughly $20,000 per year to treat this condition. And there's also associated comorbidities, including IBD. Okay, so what do we actually know about HS? Well, really the pathogenesis is unclear. And when you talk to the patients, they tell you it's the perfect storm. So we do know there is some genetic susceptibility. Mutations in gamma secretase proteins like nicastrin have been found in familial cases of HS. We know that there's a dysbiotic skin microbiome that likely contributes to the inflammation, but we don't understand how you get that dysbiosis in the first place. The predominant thought in the field is that the lesions start through hair follicle occlusion and defective support. There's this aberrant, dysregulated chronic inflammatory state. And it's not like atopic dermatitis or psoriasis where it's polarized in one way. It seems like all the inflammatory pathways are up in HS all the time. So it's like this constant fire. We know that hormones do play a role, but we don't fully understand why. And there's also these exogenous factors like diet, smoking, and mechanical stress. Okay, so my lab takes the approach of, and we actually understand what's going on if we listen to our clinical colleagues and listen to the patients. So the first thing is notice that not all these HS lesions are the same. Okay, so this is a clinical scale. This is the Hurley stage, stage 1, 2, and 3, mild, moderate, and severe. They're all kind of different. So how is the disease severity defined? And this depends on who you ask. So when you ask your clinical colleagues, they define disease severity by their clinical staging system. So like the Hurley scale, the Sartorius scale, the HiSCR, something like that. But all of these kind of resolve around counting the number of lesions, how much body surface area is affected, whether or not there's the presence or absence of tunnels or fistulas, and whether or not scarring is present. So we asked in the laboratory, well, is a patient that has stage 1 disease different from a patient that is stage 2 or 3? And what does that actually mean? Can we define the cellular and molecular signatures that underlie the clinical staging systems that my clinical colleagues use? So to do this, we curated a cohort of 39 individuals that are healthy and 87 HS patients to define this disease severity in the laboratory. So this is our cohort here. We went ahead and tracked age, sex, the Hurley stage, and the body site from which we captured the tissue. All of the demographics are down below. These are all skin samples that we got. So what do we do with the skin? We capture the full thickness skin from these patients. We take it into the laboratory. We go ahead and block some of it for histology. We perform a disc-based digestion to separate the epidermis from the dermis, and we can do a bunch of assays on those two components. And then we further break it down to understand what's going on at the individual cell type, and we've been focused on the keratinocytes. Okay, so this was what we thought was a really simple question to ask. Is skin inflammation different between a stage 1, 2, or 3? And so to take this approach, we recruited a dermatopathologist from my department. We took 44 of our skin samples here. We blinded them, and we asked our dermatopathologist to rank the level of epidermal and dermal inflammation between all of our different slides, and then identify which immune cells are present and where. Then he gave us back this ranked list. We unblinded it, and we asked the question, where do the staging systems fall? And lo and behold, here, I'm showing you some representative H&Es. If you look across the top, this is epidermal samples from healthy controls, non-lesional HS skin, and then stage 1, 2, and 3. And you can appreciate that as the stage severity increases, so does the level of inflammation, and that is significantly correlated. Whereas the level of dermal inflammation, the bottom row slide or samples, there's no difference. The dermal inflammation is constant, whether or not you're stage 1, 2, or 3. So this gave us a really interesting question to ask, like, what is going on with this epidermal inflammation? So the first thing we did was ask what immune cells are actually present. We did some IHC here. And we find that there's primarily neutrophils, NK cells, and CD8 T cells infiltrate the epidermis in these patients. And the next question we asked was, how did these actually get here? So we took our cultured keratinocytes, and we cultured HS keratinocytes, our healthy control keratinocytes, and we ran a batter of qPCR for cytokines, chemokines, and lo and behold, the HS keratinocytes autonomously secrete many more chemokines than our healthy controls. So the keratinocytes themselves are calling these immune cells into the epidermis. We next asked, what is going on in the epidermis? What is the inflammatory profile? And so here's a battery of, again, qPCR looking at a variety of cytokines. And we find that our HS keratinocytes produce more of these inflammatory cytokines than our healthy control cells. And it's all summarized here on the right, or the right left side of your screen for you. And you can see that there's a couple that are up no matter what, if you're stage 2 or stage 3, but there's also significant elevation in a bunch of these that are stage dependent, much more in stage 3 than in stage 2. So what do we do with all this information? So we put together a profile of what's going on in the epidermis, but also what the keratinocytes themselves are doing. And I think you can appreciate here that as we move through the early stages of disease, this inflammation gets far, far worse. So what does this mean for the patients? Well, we do know that HS, unlike PC, does have some FDA-approved therapies, but these are all biologics that target a single molecule. The ones highlighted here in purple are FDA-approved. But we have this broad inflammatory profile. We need something different than a single molecule. So we thought, well, we could try a JAK inhibitor. And the one that we tried is ruxolitinib. So ruxolitinib has been tried in HS patients orally with pretty good results. We asked the question, could we do this topically? Like, we know it's the epidermis, could we do it topically? Well, to proof of concept, we took our HS keratinocytes in culture, hit them with ruxolitinib, and I think you can appreciate here that with ruxolitinib, we kind of blunt a lot of the inflammatory response, but not all of it. So it's not a complete sledgehammer. So we do know that the keratinocytes and the immune cells in the epidermis contribute to the inflammation, which evolved by a stage. The epidermis is a very diverse immune cell infiltrate in HS lesions, and it's kind of marked here in our favorite diagram. We know that the keratinocytes are not bystanders. They're actually contributing a lot to this epidermal inflammation profile. And then our proof of concept study in the lab suggested to us that maybe topical drugs could work. So that's exactly what we did. We went to our industry sponsors. We convinced Insight to let us run an investigator-initiated trial where we basically applied topical ruxolitinib to patients. So here's our study design here. Because HS kind of waxes and wanes over time, the first part was just an observational period. We recruited a bunch of patients and we just asked what is going on with their normal disease flares. Then once they hit baseline, their week 0 time point, they applied topical ruxolitinib as field therapy twice a day. And then 16 weeks later, we looked for outcomes. Along with this, we captured biopsies before and after, and the clinical reported outcomes. So we were capturing the AN count, lesion counts, the HASI-R score, photography, as well as the patient reported outcomes. And these to me are the most important. So this is their quality of life and their pain scores. What do the patients actually think is going on? Here's our cohort demographics. We got 10 people to enroll. 4 unenrolled themselves during the observational phase. They were just not going to wait for the drug, but we did get 6 patients to complete the study. These are the cohorts here. They were all women. They were all stage 1, mean age of 33. They were all white, and those were the body sites affected. First things first, this drug worked. So 5 of the 6 patients had a clinical response at week 6. And so I think you can appreciate here the AN count dramatically drops off within 4 weeks of treatment. We also see that the HASI-R score, which is the clinical metric for success, drops off again by 4 weeks. And you can appreciate on the x-axis there, I list S for screening, -8 and -4. So this is all before, this is during the observational period. And what I highlighted here in the boxes, that's during the actual treatment. So clinical efficacy is working according to the clinician reported outcomes. What do the patients think? Patients think that it improved their quality of life and pain. So with the high score, which is a validated measure for quality of life metrics in HS, a score over 20 means that this significantly impacts their day-to-day living, their quality of life. Anything under a score of 14 means that their daily life is much, much better. So the way that we looked at this is we looked at the average score during the observational period and then compared it to the average score at the end of treatment. It got better. Their pain scores also got better. Anything over a score of 4 means that they have significant pain during day to day. Anything around 2 means their pain is manageable. So we know it's working clinically. The patients are pretty happy with it. What do we see when we look at the punch biopsies that we captured? So this is just a representative of week 0. So the punch at baseline versus week 16. And I think you can appreciate here, and a lot of epidermal inflammation is present beforehand, but afterwards, it basically resolves. When we quantified all this throughout all of our patients, you can appreciate here that the epidermal inflammation went way down, as well as the epidermal thickness, except for one patient. The little triangle there, that's our one non-responder. So can a topical drug applied to the top of the skin get deep enough to affect the dermis? And I think you can appreciate here with the H&Es, yes, it can. So that's the baseline, a lot of inflammatory infiltrate. By week 16, it's all but gone. Again, when we quantify how much is there, all of the responders, the inflammation went down, and our single non-responder still went up. We next looked at our punch biopsies, but this time we did RNA-seq. And so we took a very broad, unbiased look to say, what is this drug actually doing? And we compared before and after. And so I think you can show here, this is our one non-responder. They didn't really have a whole lot of change in their gene expression. Overall, we see roughly 200 genes were changed by this drug. Looking at those genes, we did a GSEA and asked, what are the top pathways? And lo and behold, a lot of inflammatory pathways are being impacted by ruxolitinib. So next, we went back to our punch biopsies and said, okay, if there's a bunch of inflammation that's being affected, can we actually see it? And so these are just some IF images where we looked at neutrophils, both before and after therapy. And I think you can appreciate here that the neutrophils do decrease. So we had 4 of our 6 patients that had neutrophils present at week 0, and those neutrophil counts were reduced in 3 of 4 by week 16. We also looked for plasma cells. They're highlighted here in the yellow. 5 of 6 patients had plasma cells at week 0 at baseline, and they were gone in 3 of 5 at week 16. All the patients had T cells present at week 0, and all the T cells disappeared in our 5 responders by week 16. So this drug is getting pretty deep. So we do believe that topical ruxolitinib is effective in mild HS. 5 of the 6 patients achieved that success metric, that high scar 50. They have decreased qual scores and pain scores, highlighting that the patients really like this drug. When we took the patients off this drug at week 16, all of them begged me to allow them to continue on this drug. We just couldn't do it because of how the study was set up. And we know that topical rux can actually blunt both the epidermal and dermal inflammatory activity. So this is the approach that we take. We talk to our patients. We ask them what are their key problems, what are their issues, and what are their concerns. We talk to our clinical colleagues to say, what are you seeing? We're hoping by defining the inflammation by severity, we can identify new novel therapeutics. And our work hopefully has shown you that both the epidermal and keratinocyte-mediated inflammation contributes to HS, and the skin is a druggable target. So can we use topical therapeutics for this condition? I don't do this alone. This is a complete team effort. So this is my lab. The 2 people, Steph and Mac, are talented postdoc and a student that really pioneered a lot of the analyses. I can't do it without my clinical colleagues and our surgery colleagues who get me the skin that we need. Thank you.
That's a fantastic talk, and this is a really huge issue for HS patients. So I'm wanting to tie HS to keratins here, and there's this disease called Dowling-Dagos disease, which sometimes co-segregates with HS, and it's a keratin 5 haploinsufficiency. Your data suggests HS may be a keratinocyte, possibly secreting cytokines, et cetera. Is there some way we could connect what's going on inside of the keratinocytes with this sort of homing mechanism? Is it really not an inflammatory disorder, but more a primary keratinocyte disorder? I'm wondering what your thoughts are on that.
I agree with you. In short, yes, we don't fully understand what is driving HS, but I mean, most of the field believes that it starts with the hair follicle. The hair follicle is comprised of keratinocytes, right? So if I was to place a bet, I think the keratinocyte is one of the initiating cells. Whether it's intrinsic or if it's responding to some type of signal from the environment, I'm not entirely sure. And it's so complex to try to tease some of that out. But we do see dysregulated keratins in all of our punches. And the strange part is it's very different patient to patient. So if we look carefully at what is a stage 1 or stage 2 or stage 3, we kind of think that all stage 1s are kind of this and stage 2s. No, every single one of our stage 1 patients are truly unique and where they're starting from. And so I don't know if we need a better staging system or we just need better classifications, but keratins are dysregulated. I can't find the smoking gun yet, but I do believe you.
Can I just following Corey's remark, actually there is a good reason for having you here. It's because actually a large portion of patients with PC and K17 mutation have something which actually from a clinical point of view cannot be distinguished from HS. And so, and there are also additional similarities. So for example, something which is common to HS and also those patients who have K17 mutation is aberration in notch signaling. And there are also some data suggesting that JAK signaling is actually mediating the beneficial effect of statins in PC. And taken all together, this actually make the case once again, or maybe trying JAK inhibitors, not only as a way to downregulate immune responses, but actually, just as Corey was mentioning, maybe in order to actually downregulate the aberration which are leading to this keratin directly related disorder. I think that there is a lot to be learned from this. The only problem, and maybe we can speak about that later on in terms of strategy, is that what works on intact skin or more or less normal skin in terms of penetration and delivery may be more challenging when speaking about a skin which is affected by PC, but that's another issue. Thank you.
Thank you, Amanda. Beautiful talk. Just quick question. Is netosis a driver of disease, either presentation or severity?
Yeah, great question. So Angel Bird's lab has been studying netosis in HS for many years now, and she has shown that there are presence of nets in these patients, and they do increase with severity. And she has found through her work that there's actually mutation in one of the DNA enzymes that helps break down the nets. So there's patients that have this mutation, so they're unable to clear them. And so it kind of acts as this constant inflammatory environment in certain patients, but they're not present in all. And there's really no rhyme or reason to understand like why they're there or not, right? So like we really don't understand. We can't lump them all together yet. But yes, it is there in some patients.
Thank you so much. If there are any other questions, I'm sure we can discuss more at lunchtime. Thanks, everyone.
This is a perfect segue to the next session. And as we're going to be focusing on therapeutics. I'm Joyce Tang. I'm a professor of dermatology and pediatrics at Stanford University. I've participated in many clinical trials in both ichthyosis, PC, and other rare genetic diseases. Lots of heartaches and lots of hopes as well. Just as Janice said, together we will find a therapy, and together we'll rise. Without further ado, so the next session going to be both from gene therapy to drug delivery, as well as some of the exciting pharmaceuticals. So our first speaker is Dr. Marta Garcia Diaz from the Carlos University of Madrid. She's an associate professor of biomedical engineer, and we're going to hear from her about CRISPR and gene therapy. Yeah.
So thank you very much for the introduction. So for many years, our laboratory has been involved in the development of different platforms to study the molecular mechanism of different skin diseases. And for that, we developed this skin humanized mouse model, where from a small biopsy, we isolate the fibroblast and the keratinocyte. We grow the cells in culture, and when we have the enough number of cells, the fibroblasts are embedded in a fibrin matrix coming from the blood the plasma, generated the thermal compartment of the skin, and on the top we put the keratinocyte generated this dermoepidermal equivalent, which are then grafted onto the back of the immunodeficient mice. And once this equivalent take and mature, we obtain a human skin with the same characteristic of the original skin. So one of the advantages of this system is that from a small biopsy, we can generate big areas of skin, transplant different mice, and then make all the studies in a homogeneous manner. So with this methodology, we be able to modulate different skin diseases, among that Pachyonychia Congenita. So once we have the model, the next step is try to correct the defects associated to each disease. And for that, we are developed a preclinical gene editing protocol for ex vivo and in vivo correction of this disease, working with CRISPR-Cas system as gene editing tools due to the fantastic characteristic of this system. So our model allow us also try all these tools, both ex vivo, modifying first the cells and after that transplant to device, or in vivo, try all these tools in our mouse model. For genetic editing strategy, we need to take into account the pattern of the inheritance. For dominant negative diseases, like is the case of C, the strategy should be aimed to inactivate just the mutant allele. And CRISPR-Cas is very efficient when the mutation generates a PAM. PAM is a specific sorry. Okay, it's necessary to activate the CRISPR-Cas system, okay? So once the CRISPR-Cas system edits the cells, the subsequent mechanism of the cells by no homologation joining are going to induce indels. These indels are going to disrupt the normal correct translation of the genes, and as consequence, we are going to inactivate the mutant allele. No synthesis of the pathogenic protein are going to be doing, and the health proteins can do the function. So for this reason, we think that gene editing allows permanent correction by inactivating the mutant allele when and if we are able to correct the stem cells that are localized in the basal layer. So we try the strategy first in ex vivo models in cells coming from a patient with this mutation in the gene of the keratin 6A, and this mutation generates a PAM for keratin 12. So then we generate guides complementary to the sequence adjacent to the PAM, and by nucleofection, we introduce all the RNP complex inside the cells, and these are the results. We can see at DNA levels the addition, and when we do the immunofluorescence against keratin 6, we recover the normal filaments of the keratin 6 compared with the characteristic aggregate of the PC in this the PC. So we move all to the ex vivo therapies, and before to do the analysis, we induce the hyperproliferation state characteristic of this patient by tape stripping. And these are the results. A short time, in all the cases, the wild type PC and PC edited, we can see the acanthosis in all the cases with concomitant expression of Cas-C in response of the hyperproliferation we made by tape stripping, and a lot time enough to resolve the damage we made. Just the wild type and the PC edited cells are able to recover the normal phenotype of the skin, while the PC retain this characteristic of hyperproliferation and PC expression. So once we see that the system works, we move on to the in vivo analysis, and for that, we work with herpes-dependent adenovirus due to the all these characteristics. One of the more important one is the low toxicity in vivo compared with the first generation adenovirus that was used before, and to generate the particles in the vector, we clone all the units necessary to generate the CRISPR-Cas system. The guide is the guide that we used before, the gene of the Cas12, and we couple ERS-GFP that allowed us follow the transduction of the vector. So we try the particles in the same cells as before, and this is the result. At DNA level, we see the addition again, and if we see the filaments, we recover also the normal filaments of keratin 6 compared with the aggregated of the PC cells. We move on to the in vivo analysis. Again, before we apply the vectors, we generate the tape stripping in this case for two things. One, to inhibit this hyperproliferation, and to take off the stratocorneum, because if you don't take the stratocorneum, nothing are going to go inside. And also we use microneedles to generate microholes in the epidermis and allow the adenovirus reach the basal layer where the stem cells are localized. So these are the results. Again, at a short time, in all the cases, we see this hyperproliferation stage, and a lot of time, it's true that not all the area treated with adenovirus recover the normal stage of the skin. But if we see the immunofluorescence, we see that the parts that that the part that recover the normal stage don't express keratin 6 anymore, while the zones that are not recovered express also the keratin 6. The good news is that we can see in this slide that the vectors are able to transduce the basal layer where the stem cells are. So as was expected, at different times we need to treat the patients with the adenovirus, and here is where problems becomes. Why? For the immunological problems that have this virus. It's true that working with the hyperdependent adenovirus, all the problem of the toxicity is gone, but still we have the immunological problem. So we designed this experiment to see the numbers of the antibodies against the adenovirus. So just at the second doses of the adenovirus, we start to see the antibodies. So to try to improve this number, we try with different immunosuppressants. These are some of the immunosuppressants that the other research use with the same kind of vector that we are working on. And we repeat the same experiment with cyclosporins, and these are the result. And the good news again is with cyclosporin and without cyclosporin, in every cases, we see the transduction of the cell. It's true that with the cyclosporin, the efficiency of the transduction is so much better than without cyclosporin. So we want to repeat these experiments longer and with the different doses, and let's see what happens. So new approaches that we are working on also as improving these tools. For example, we are working in a new platform to work with helper-dependent herpes virus due to the low profile or immunoproblems that the vectors have. Regarding the no viral vectors due to the high profiles of the biosecurity. And regarding to the new technologies, we are working with the prime editing that allow us treat different mutation with the same vector. And this is just an example. For example, these are all the mutations that are described for keratin 6A. This is the gene of the keratin 6, the exon 1. This is the mutation that we are treating right now. So with the vector that we are doing now, we can treat this only mutation, and with the other system, we can treat all these mutations with the same vector. So we think that is, at least we need to try this system. And that's it. So with this study, we really believe that the gene editing techniques could provide a long-term curative therapy to improve the patient quality of life. And of course, thank you in special to Janice and Holly for all the support that you give us, and for sure, all the collaborators that working together, we can do all these experiments. Thank you very much.
Thank you so much, Marta. We had a Keith.
So really beautiful and inspiring work. But I think one of the things that's unique about EB is you have incredible selective advantage for corrected clones. And you also have a very long-lived protein that's produced. So what do you see as the critical barriers that need to be overcome to make this possible for a genetic disease for which we don't have selective advantage of revertant clones? Well, but is related to your work. I think that's the central question.
I get that everything would be corrected because everything are going to coming from these stem cells. Yeah. Mm-hmm. Yeah, wow, this is the point. I don't know the answer. We try to correct the more stem cells we can. And I guess that correct all the area that you are treated is possible to correct this area treated.
Maybe another really short question. Marta, what is the transfection rate? How many cells are the vector can get in? Ratios of transfected, not transfected.
The final target is the stem cell that are in the basal layer.
Yeah.
And if you see the immunostaining, all the basal layer in this case are transduced. So we hope that some stem cells were there, and we target that efficient.
Great. All right. And we're going to move on to the second speaker, Joshua Tan, from Assistant Professor of Biomedical Engineer from the Wellman Center, MIT, also a faculty of the Harvard MIT Joint Program. We're still looking forward to our topic on the drug delivery.
Thank you so much. I want to take a minute to echo something that Pierre said at the beginning of the meeting about the patient support meeting that occurred here the first couple of days, the last couple of days. I had the privilege of joining that meeting, and I think we all love science in this room. That's why we're in this business. But there's nothing quite as motivating and really informative as being able to sit next to someone who's living with this condition their entire lives and learn about their challenges and also and the resilience of these patients. So I'm looking into drug delivery for plantar skin, and this is a relatively new area for us. We've only been doing this for a few months. So what I'm about to show you is not a complete story, but I'm excited to show you our findings and also plans for the future. And I'd love to get some feedback from this group. Yes, it works. So the motivation really came from studies like this that was done by Dr. Leishman and some others in this room. This was a study using an siRNA to treat PC, and the treatment seems to work reasonably well. This is the area that got treated, and you can see the callus is cleared up really well. But the intradermal injections that was used to deliver this stuff was extremely painful, and that was the main problem with this approach. And this seems like the kind of thing that will keep rearing its ugly head, for any kind of topical therapy. And I happen to have some background in looking at drug delivery in skin, so I want to look at this and see if we can make any headway. So just to quickly go over some of the challenges, this is an H&E of just abdominal skin in human. I've highlighted the stratum corneum up here in the screen bracket. It's about 10 to 30 microns thick in most parts of the body. And as most people here probably know, this is our primary barrier and the rate limiting step for certainly water soluble formulations. Over here is just normal human plantar skin. Again, the stratum corneum is highlighted. In this sample, it's about 700 microns, and it's not unusual for this to be over 1 millimeter thick in the weight-bearing parts of the foot. So you can just imagine the sheer distance that a topical medication will have to go through. What you can't see very well on the H&E is that the chemistry and the diffusion characteristics are also very different in palmoplantar skin. Because of time, I'm not going to belabor it, but a lot of our concepts about drug delivery in non-plantar skin don't necessarily apply to plantar skin. So things like lipophilic versus hydrophilic formulations and stuff. The water diffusion constant in this type of skin is about 100 times higher than over here. So whereas the diffusivity is the main challenge in non-plantar skin, the distance really is the big problem for plantar skin. So a lot the approach we're taking are mostly centered around trying to bridge that distance. So, and then just a quick word about model systems. This is mouse, human, and pig plantar skin shown side by side at the same scale. Pig and the stratum corneum is highlighted with the arrows. In pig skin, it's quite a bit thicker than humans. I don't know if it's species difference or because we wear shoes and pigs don't. It's probably both. But it's at least in the same ballpark in terms of thickness and kind of morphology. And if you consider it in PC, because of the calluses, this is even thicker. It's probably getting close to where pig skin is. Whereas in the mouth, even the weight-bearing part of the plantar skin, the stratum corneum is only a few tens of microns. So I think whereas, as we heard today, mice are really important for understanding the molecular mechanisms behind these diseases. I think in terms of the physical behavior of drug diffusion, I think the pig skin and human skin are good models, whereas mouse might not be. So here's a cartoon of the skin. You notice the target for PC is the viable epidermis. That's where the mutations are. That's where you want your drug to go. When you do an intradermal injection, like it's been done before, you put a big bolus of drug straight into the dermis, and that's really, I think, the root of a lot of problems. That's where most of the pain sensation comes from. It's also very inefficient because some of the drug will diffuse up to the viable epidermis, and that where it does this thing. A lot of drugs the drug will either go, either stay in the dermis, get washed out by the blood. It doesn't go where you need it to go. So you're kind of forced to use a larger volume because a lot of it goes to waste. And the increased volume itself causes a lot more physical stress in the skin, and so that adds to the pain. I think what you really want is what's depicted here, where you have maybe smaller volumes of drugs, but concentrated around the viable epidermis and spread over a larger area. So that's also what we're trying to achieve. So we're mostly looking at two different ways to do this. The first one is laser-assisted drug delivery. This is something I worked on before. That's what I alluded to at the beginning about my experience with drug delivery. So there are two main ways to do it. One is full field ablation laser. That's where you take a laser to just blow away large areas of skin. The other way is ablative fractional laser, or AFL, where you use a focused laser beam to make these narrow channels in skin. These are commonly used for cosmetic reasons. So people try to reduce wrinkles and sunspot. This is an image I just found it on the internet, so from some clinic that does this, just to show you what these lasers are usually used for. For our purpose, we do it a little differently. Ideally, you try to get rid of most of the stratum corneum, but kind of stop at the viable epidermis, and then apply the drug so that it has direct access to your target. So the big question there is whether we can actually do this, because that's not how these lasers are usually applied. So this is one example. In the pig skin, we're using an erbium YAG laser at about 5 joules per centimeter squared. For most people, that number probably doesn't mean a whole lot. We just basically crank the laser all the way up because it's very thick skin. And even at that setting, because it's so thick, you need multiple passes of this laser to get down to the depths you want. So this is after 10 passes, you get a little under 1 millimeter of ablation, and your target is down here. So you've still got quite a while to go. At 20 passes, we get about 2 millimeters. That gets you close to the viable epidermis. And then at 30 passes, we're now at about 3 millimeters, and now you're well in the viable epidermis. So what this shows is that we can use this method to get direct access to the viable epidermis, and there's roughly a linear relationship between the number of pulses and the ablation depth, which is a good characteristic to have, because then if you know how much you need to, how deep you need to go, you can kind of count the number of passes and get there, and that makes it a more practical treatment. So I'm going to switch gears to a different modality we're looking at, and these are needleless jet injectors. These are injectors that propel the formulation at a very high speed at the skin, so that when it hits the skin, it pierces the skin and deposits it inside. So I like this because you can get very good consistency in delivery depth, and generally it's less painful than needle injections. Not always, but I think if you control the parameters right, you can get less pain with the injections with it. This is not a new technique. There's some splashback always, like not all the drug comes back. Some of it not all of it gets in the skin, some of it comes back at you. And this was used for mass immunization in the 70s. But it turns out because of this splashback business, some of the blood and fluids come back into the applicator, and then when you use the same applicator across different people, that's an excellent way of spreading infection. So, so jet injectors kind of went out of favor for a while, but I think they're coming back now because some of the improvements in technology that I'll discuss a little bit. So unlike in the laser where we have a very well-defined kind of morphologic endpoint we can use, these injectors don't. So we had to figure out a way to see where the stuff went, and we actually had to experiment quite a bit to find a solution that works. But finally, we settled on these histology marker dyes. So we take the dyes, we had to dilute them a little bit to get the right viscosity, but then we load them into different injectors and then try them in human plantar skin that we get from amputations. So this is first we tested in pig skin first because it's easy to access. This is typically what it looks like with an intradermal injection. Depending on when you look, you can see the needle along the needle track, all the green dye being deposited. This is one jet injector that got pretty deep into the dermis. It's probably deeper than we really want. But this is another jet injector setting where you get pretty superficial deposition. And then we also tried this in human plantar skin samples. This is one example of what that could look like. Up here, you can see the channel that's created by the jet. And then down in the dermis, you see all the green stuff. This is another example of kind of the result that can come out. At first, I thought this was a sectioning artifact, but then when we look more closely, along this space in the middle, you can see the green dye along kind of marking the edges of this. So I think this is when the jet deposited in the middle of the epidermis and kind of sheared the tissue, because there's no extracellular space for the stuff to go. So it looks huge here. It's about 5 millimeters wide. So that might be more volume than you want, but that's something that you can change. And I think in terms of the depth of deposition, this is kind of right on, because this gets you right at the target. So so we're right now going through kind of characterizing a lot of different injectors and delivery methods to see what that looks like and see what's the best way to get the stuff to where we want it to go. But the injectors that we've tested so far are commercially available, and there are also older generations of injectors. So the jet is mostly generated by either compressed springs or pressurized gas, and these are you get what you get. Like with the injector, you have no control over it. You can't go in and change the compression constant on these springs. But there are newer versions of injectors now that use different technologies to generate the jet, and these give you a lot more control over kind of how the jet is generated and how it gets deposited, how deep it goes, the volume, and things like that. The other advantage of these newer generations is that the ones we've tested are like needles. You take a bolus and you put it into the skin. There are newer injectors that work more like inkjet printers, so you have smaller volumes and you can kind of spread it over an area and define where you want it to go. So these are the next ones we like to test. These aren't commercially available yet, but there are a number of companies developing these, and we've just recently finished the legal stuff so that we can get our hands on this and test this. So we'll be working with these very soon. So far, everything I've told you has to do with the kind of physics of the deposition, whether we can get the material at the depths we want. But I also wanted to take a look at function. And something I really appreciate about the PC project is that we're a very collaborative bunch. And I feel like I can say we now. It's since I was here last year. Well, I talked to Janice about this, and she put me in touch with Robin, who was really helpful and gave me a lot of very good advice. One of them is to get in touch with the RNA Therapeutics Institute at UMass, because they have some of the best in class RNAi technology. So I've talked to them, and we came up with a plan. We're going to keep using the post-amputation human plantar skin as our model. We're going to establish the delivery method like we've been doing, and then they're going to give us some model sRNA to test the effectiveness of the delivery method and also the ability to suppress gene expression. And then if that works, then they're going to help us with some sRNAs against the keratins that are the targets in PC. So that's the plan. Hopefully in maybe a year when I come back, I can tell you some results from that. With that, I'd again like to thank the PC project and also my colleagues and collaborators and advisors who've helped me with this. Thank you.
Joshua, any questions from the audience? Sensi.
I'm really wildly excited about this for obvious reasons, so that's good. But I wonder, have you, as you mentioned, the limiting factor was really the pain, and I know you've probably talked to Jan about that before, and it was a sort of debilitating kind of pain for, it wasn't able to be used for that reason. And so I'm wondering, have you really tried on a PC patient? And if you haven't, I would encourage PC Project to think about actually getting a patient up to try the laser in person, really, before you spend a huge amount of effort going forward. Just try it and see, how much pain, if any, there really is in a PC patient, because it's a different neurovascular structure. It's a different, and I just think it'd be worth it, because if it works, great, you do full steam ahead, right? If it doesn't, if it's painful, then we may need to rethink it.
Yeah, the neurovascular abnormalities inside the callus is something that I just learned from this like a couple of days ago at the patient. In normal, like the rest of us, there's no sensory function in the callus. It turns out in PC. So whether you can, how to do it without a lot of pain, I think that really is an important consideration. At least the people I've talked to seem like I think PC patients are really good at trimming their calluses and at doing it where it's to the point where they can tolerate, but not further, because they do this their entire lives. So I think combining that with so I didn't go over the fractional laser. If you just try to get through a thick callus, it's not, because you have to take so many pulses, you can't do it. That's why I didn't have any data on the fractional laser. But I think if you pair the callus to a reasonable level and then use the fractional laser, because the amount of ablation is a lot less, so that might be a better strategy. So I think there's some of those tricks you can play, but I get your point. That is the big hurdle.
I completely agree that I use erbium laser almost in every one of my clinics, and the energy you can dial up and dial down, and so maybe even consider the first, and also that the pig skin is really tough because the keratin, so that Joshua has to do 20 pulses, and you really don't have to get to that depth with the human plantar skin. and maybe a couple of, just one low energy or two low energy pulses, and to put the numbing on first and facilitate that penetration and before you're trying to deliver a drug. So we should really put our heads together to come up with a feasible clinical protocol for these. Great. There's another question back there.
Yeah, Jack, administrator at Wake Forest. I thought that was an awesome presentation. Just curious if you've seen how some of our, colleagues in hyperhidrosis and psoriasis have used iontophoresis for plantar pulmonar drug delivery, including, I think, methotrexate iontophoresis. And I'm wondering if that's a modality that could be combined with keratolytics as sort of painless options, and then it might reduce the amount of pulses, necessary to then use laser and maybe combine the three, something of that nature. And iontophoresis, it might be limited by the charge of whatever the molecule that we're trying to treat with, but.
Yeah, that's a good one. I think, like you said, there's the consideration about the charge and size of what you're trying to deliver to see if iontophoresis could work. Combining with keratolytics, that's another thing I learned recently, is that if you mess with the physical characteristics of the callus, that alone sometimes could be very painful, like in the ways that certainly I didn't expect. Like I talked to someone who said there was a clinical trial drug they tried that the formulation softened the callus, and which I immediately thought that would be awesome. And he's like, it made him miserable, because then when he walked on this, it was hurting his feet for weeks afterwards. So like Sanse was saying before, I think we've got to tread carefully, but we'll just have to figure that out.
And also it takes much, much longer to do iontophoresis on the fractional. Wonderful. Thank you so much. Thanks again, Joshua. So we're gonna switch gear and move on and talk about some of the newer pathway and pharmaceuticals in the next two talks. So our next speaker is Dr. Karen McGuire. She's a PhD in bioengineer as well as molecular biology. She's also the funder of Biomedics, and we're so looking forward.
Thank you, Joyce. I appreciate that. And thank you again to PC Project and the organizers. I really appreciate you guys kind of last minute slotting me in here. We just managed to get some of this preclinical research done, and I think it's exciting enough to share with everybody. So I founded Biomedics back in November of 2014. We're primarily privately funded. Our lead indication for our small molecule mTOR inhibitors is actually epidermolysis bullosa simplex, which we recently completed a small pilot phase 1/2A clinical trial with the team out at Stanford, and we're fingers crossed any month now we will be ready to start our phase 2 clinical trial in EBS as well. And so we've kind of started to go back and look and see, well, if this drug is working in EB simplex, perhaps there's a application for it in both PC and epidermolytic ichthyosis and some of these other palmoplantar keratodermas. So for those of you that are not familiar with mTOR inhibitors, I'm overly familiar with them because I started out in oncology. And at that time, mTOR inhibitors were quite popular. So a lot of these had problems when we went to use them in oncology, right? So they're very potent, they're high molecular weight, or they had issues with rapidly converting from an active to an inactive isomer, for example, stilbenes and flavonoids. Or you had the rapalogs, which also have immunosuppression issues. And overall, most of these are either too toxic or poor bioavailability. So what we decided to do is go after and take those stilbenes and actually turn them into substituted tolins. And so the benefit of these substituted tolins is that they're actually low molecular weight. They're under 500 Daltons, so they can easily penetrate the skin, right? So not a problem there. We get them in, they're also slightly polar. They tend to stay where you put them, right? So you don't really get much in the way of uptake into the bloodstream from there. They're super stable, bioavailable, and they're good from a commercial standpoint as well. Easy to synthesize, easy to make, they're quite cheap. And so since making them, we've put them through toxicology studies. We've had them in about 100 patients so far, primarily putting them in open wounds. So we did a bunch of wound repair and tissue regeneration studies with Rob Kersner down at the University of Miami. So how do we think mTOR inhibitors could be working in PPKs like PC or in a disorder like EB simplex? So when inhibiting mTOR, specifically with these types of inhibitors, we're able to get all the way down to the activation of autophagy. And so autophagy's primary function is really to sort and remove any kind of damaged or aggregated, misfolded, mutated proteins in general. And in this case specifically, we think it's removing a lot of the aggregated damaged keratins that are present. You can actually see this in EBS cells. Aggregated keratins can even still hang around after the cells divide within that basal layer. So I finally managed to get some PC cell lines. So I think Len Millstone, we decided he's not here, had provided these cell lines to Keith Choate. And we were able to get a hold of a few of them with a very nice one with a keratin 6A mutation, one with a keratin 16 mutation, both in that kind of key 1A domain that's essential for assembly. So we then took these cell lines and we pre-treated them with our lead compound here, which is BM3103, and with a dose escalation of rapamycin. And we could not get rapamycin to induce autophagy in keratinocytes. And this is not unique for us to PC cells. We saw the same thing in EBS cells. And we've also seen the same thing in epidermolytic ichthyosis keratinocytes as well. So when we went to we kind of quantified this out, and we were able to get a statistically significant increase in LC32, which is the kind of the endpoint for the production of the autophagosome across both. It didn't matter if it was PC1 or PC2. For those of you that are familiar with trying to measure autophagy, you can't just measure one or the other. You're measuring autophagic flux, right? So that's the ratio of LC32 to LC31. And so here you can see, again, statistically significant fold increases in this flux ratio. So the next thing we wanted to do is try and see if we're pulling these kind of bad versus good keratins out of circulation. Does that increase in these more normal K6A keratins or K16 keratins actually cause the cells to be more stable under stress? So we cultured these cells, then applied the drug for 24 hours, heat stress them. So just increase the temperature to 43 degrees C for 30 minutes. This whole heat stress assay was actually developed by Birgit Lane. And then we let them recover and stain them for keratin 6A. So as you can see, prior to heat stress, there's just a few collapsed keratin intermediate filament networks that are visible. So this is pretty common. A lot of times, whether it's PC keratinocytes or it's EBS keratinocytes, they can look under culture quite normal, right? And it's really hard to tell that those networks are fragile without, again, stressing them in some way. And this can be done using heat, osmotic stress, chemical inducers as well. And so after we pre-treated with the drug for 24 hours, we were then able to almost restore the keratin filaments. So they're almost back down to baseline levels without heat stress. So sort of in conclusion, we're trying to still figure out what is it that makes PC keratinocytes resistant to rapamycin-induced autophagy. I'm sure there's some great molecular biology labs that would have the time to do this. We don't really have the time or the resources for this one, but I think it would be really interesting to know. But that the drug does seem to reduce mutant and aggregated keratins, strengthening the keratin intermittent filament networks. And ultimately, this will have to be proven out in the clinic, right? And so my challenge, what I would like to talk to as many people as possible today is clinical endpoints for PC. Pain really didn't pan out for us very well as an endpoint. So we need either better pain scores, we need to start looking at some way of measuring the callus, et cetera, because I would love to run a PC trial, and I'd love to have some good endpoints to do it with. So go ahead. You're good. You're good. You're good. Feel free. There's a follow-up. Feel free to reach out. We do send out a newsletter if anybody's interested in kind of following what we're doing with EB and PC. So thank you.
Any questions from the audience?
Cell lines and eczema. Both.
No, they're not easy. I don't believe so. Those are TERT modified. Yes.
Yeah, sorry, Karen,
Yeah.
the exposed cells, was this, meant to mimic systemic, or was it applied, the gel topically? And I'm just wondering if someone took oral rapamycin.
So oral rapamycin can be quite effective, right? But it's not well tolerated in EB simplex.
Okay.
I don't know in PC.
Gotcha. Thank you.
Sure.
I mean, it's as more of a cell biologist who thinks about autophagy, I'd love to do some sort of work with you to look at like what's happening at the molecular level to the aggregated keratins. Can we track them? Is there keratinophagy? This has been talked about by other scientists. Are there specific receptors that tell the cell to get rid of this misfolded keratin? And there's a lot of cool cell biology that might allow us to taper in your therapy even more specific if we're worried about side effects of something like rapamycin, which is not a benign drug to take orally.
It is not a benign drug to take at all. And we have been able to kind of co-localize those keratins into the autophagosomes, but only in the EB simplex cells, which I didn't really show you the data for here. And we did actually some electron microscopy work as well at the end of our study to look at, the status of the keratin intermediate filaments after 10 weeks of topical administration.
Yeah, so I couldn't agree with you more about needing really good clinical endpoints. And we did a really nice roundtable, some in-depth discussion about endpoints with Palvella when they were starting to do the rapamycin. And I would suggest that it might be a really good idea to get their data. Joyce was there with me, and there was a lot of clinicians. And one of the things that I would encourage you to think about is that one of the recommendations that I made at that time, and I think there were a lot of us who thought this was a good idea, is to actually look at the functionality that was increased in the patient. Because the thing is, each patient really pushes to their personal threshold. And so you're not going to really get pain as a good measure, I don't think. I think what you get as a good measure is how much they can do before they get to that threshold, right? And so back then they didn't have such great wearables, but we've got great wearables
Correct.
now.
Yeah, and Janice and I have been talking about some of
Yeah,
those great
and
wearables, yes.
so I think that's an example of some of the things that you would get from a nice little round table. And PC Project might even be able to arrange a similar round table type virtual round table kind of outcome measure thing, maybe, I don't know, Jan, you have to say, but I think it would be a great brainstorming session and fun to do.
Absolutely. I'd love to talk paracalluses first, don't paracalluses first. I mean, I think there's a lot of ways we could go here, but I will let you guys get on with your agenda.
Thank you so much. We're running a few minutes behind, but it's a great discussion, all worth it. We are going to move on to the next pharmaceutical talk and TRP3 inhibitor, and this is by Dr. David Everazzer, and he is the CEO of Kamari from Israel. Also the associate professor of biotech department at the Bar-Ilan University. Welcome.
Thank you, PC Project and Janice for organizing, all the organizers for inviting me, and thanks, Joyce, for the introduction. Being the last speaker, we don't need clinical trials to know that PC patients, researchers, and clinicians probably get a bit edgy before lunch, so we'll try to focus on the most important points. So TRPV3 is an ion channel which is highly expressed in the skin, and I think the most important biological phenomenon is that it has been directly linked to a well-established skin disorder, Olsen syndrome, which was identified almost 100 years ago. But we know that a mutation in this ion channel results in a clear and very specific readout in the skin in a very clear disorder. We'll touch about this in a moment. So therefore, identifying a target that has a direct biological effect is a very important step in the drug development process. Now, we know Olsen syndrome is a severe and life-threatening disorder. It has it's a rare, even ultra-rare phenomenon. Nevertheless, the biological effects, the clinical effects that we see in this syndrome are shared in a large number of different keratoderm. Because there's no clinical treatment, direct clinical treatment for this disorder, we found, and we see this as a very clear indication that, yes, this is a target we can go after and use this as a role model for the broader development we'd like to move forward. So we have identified a number of very specific TRPV3 inhibitors. These are all small molecules, classical drugs with IC50 in the single nanomolar range. They're all very, they're safe, and importantly, they're highly selective. So we have two classes, topical and oral, and we'll try to address both of them in the next few. Now, we know that TRPV3 is running through the EGFR pathway. 30 years ago, when I was doing my PhD at the Weizmann Institute, and we first identified, it was in tyrosine kinase inhibitors to FGF and EGF receptor. There was this notion that by this non-selective inhibition, we'll be able to solve all the problems that involve these pathways. Unfortunately, this has not been the case, not for cancer and not for other indications. And the need for a very both selective and safe and tissue-specific inhibitor is well established now. And that's really what we are trying to do. Go after a clear target that is safe, selective, and tissue-specific. So looking at, for instance, this is a a PC skin model. When we look at in this case, this is our oral TRPV3 inhibitor, we can see that in a dose-dependent manner, we can reverse the skin both from we can inhibit unnormalized proliferation and normalize the cell differentiation as well by this specific TRPV3 inhibitor. Furthermore, when we went on to in vivo animal models, and in this case, this is the gain of function mutation knock-in mouse, we can see once again in a dose-dependent manner that by treating these animals with our inhibitor, we can actually, first of all, lower the amount of scratching bouts. And furthermore, when we look at additional symptoms, we can normalize the skin compared to vehicle. And furthermore, looking at different parameters of keratoderma, we can actually, once again, in these animals, reverse the manifestation of the gain of function mutation. So all this has strengthened the notion that, again, either in a topical or an oral treatment in this mouse model, TRPV3 seems to be a clear and promising druggable target. So we initiated a clinical trial, a phase 1b study in patients. And what we've done here is treating, was using our topical drug, our topical TRPV inhibitor. And we were able to show that, first of all, we can demonstrate a very good safety profile with our topical TRPV inhibitor. And furthermore, when we looked at the efficacy results, and we've had several cohorts of the study, including an extension study for 16 weeks, we can see that 85% of the patients have responded to treatment in the different scores that were measured. And we can see, and I'll show you in a minute in more detail, we can actually see a shift and an improvement in the different scores. And furthermore, also measuring pain, which is, as mentioned previously in several discussions this morning, is another critical aspect of PC, but we were able to see an improvement here as well. So a little data, we don't have too much time. We can see a very clear shift when we measure patients in the beginning of the study, who most of them were suffering from very severe or severe pain or severe disease, for instance, in the IGA assessment score. After fulfilling the full study, a vast majority of the patients are now only in a moderate stage. We have no severe, very severe patients, so we can see a clear shift in the IGA score of these patients throughout the treatment. Talking about pain, for instance, when we look at measuring the VAS score, once again, at the end of the treatment, we can see a reduction in pain of at least 10 points, in 80% of the patients treated with our topical drug. So we definitely feel that TRPV3 inhibition is a valid and important target for treating these disorders. And once again, PC is as you all well know, is not a TRPV3 mutation, but rather an overexpression. Nevertheless, its involvement in the different parameters of the disease is clear, as we can see in the shift following the treatment. So with that said, we have also developed an oral TRPV3 inhibitor. Now we understand that having an oral treatment has several different advantages. First of all, we're able to treat, the administration is easier. Pill taken daily will always be easier than having to apply a topical cream. Furthermore, having a systemic treatment throughout the whole body, we believe will increase the efficacy of this treatment. So as mentioned, and we will hear more about this tomorrow, we've demonstrated that also our oral drug is showing efficacy in both in vitro and in vivo models of the disease. So we feel quite confident that taking this oral drug is definitely another valid approach we would like to take. And we are now approaching the clinical development stage. We will initiate a safety study in healthy volunteers as the first stage with our oral drug. Hopefully this will happen very soon, the next quarter. And immediately thereafter, go into Olmsted patients with our oral drug. Obviously, all Olmsted patients, as the hallmark of TRPV3 involvement in disease, are a very important patient population, despite the fact that an ultra rare patient. And immediately thereafter, we plan to initiate a clinical study with our oral drug, hopefully based on positive data we will be generating, also in PC patients. So, summarize, we think that TRPV3 inhibition is a valid drug development target. We're very encouraged from the clinical data we've generated so far and from the preclinical biological data also for our oral drug. So I'd just like to end by thanking all our collaborators at the PC project, Janice and the team. Of course, our clinical collaborators, some of them are here within the room, Ellie and Adele and the who are on the Zoom. And of course, additional collaborators going forward with our clinical development are very welcome. We're privileged to work with the patients and with you as clinical treaters, the scientific community in the field. Thank you very much.
So David, any questions from the audience? I have a question about the oral inhibitor to mouse model. What kind of side effects have you noticed? And also, would you consider give it to mouse before they're born? I'm wondering whether it changes in the epidermal differentiation and maturation.
So we haven't seen any clear side effects given in the mouse model. I can say already that we were very far in preclinical development for safety, both in rats and in pigs towards the clinical trial, and we haven't seen anything either. So we're quite confident so far that the safety profile for the oral drug is very good. Your suggestion is very interesting, and we have not tested it. This is something we are definitely considering. It's a bit more complex, but this is definitely something that is worth thinking about at the next phase of development as going forward in development. Currently, we first want to focus on treating adults. Obviously, also the clinical study, the first phase will be focusing on adults, but it's definitely a valid approach that is worth considering. Thank you.
Thanks. I don't see any hands. Maybe a very last question about the pain oh, sorry. Standing right here.
I was just wondering, with some of the other trypsin blockers, thermosensation and the ability to form from noxious was an issue. And I'm wondering if in the cream, if you did any sensory testing to look at ability to detect warmth given these thermosensors.
So I'll answer your first question, and I'll hand it over to Leo to answer the second question because I don't know. These molecules are highly selective in several magnitudes of order compared to the other TRP. So we know this is not. We don't expect this to be an issue in the practical treatment, and we haven't seen anything neither in clinical trials or in preclinical evaluation. For the second question, have we tested the cream? Frankly, I don't know.
We did not test specifically for heat effects, but in testing for preclinical safety, we did test a little bit with pig skin, with mini pig skin actually, to see if there are some changes or some effects, and we didn't see any. So for efficacy, no, for safety.
Thank you very much.
Thank you. Hey, thank you, everybody. This is the end of our regular presentation part. We're going to start session 5 in 10 minutes. So this is some instructions for you. I'd like to excuse Pierre, Ellie, and Robin. You go get your lunch because they're going to be our moderators for the next discussion. So did everybody take good notes and you're ready for our big discussion over lunch? Okay. So what I want you to do is just take 10 minutes. If you need a bio break, get your lunch out there. I would like to just ask kindly if you registered for our meeting and you know if you register for our meeting because you have one of these, would you get your lunch first? If you didn't register, I think we counted and we think there's plenty of lunches for everybody and you're all welcome, but let the people who registered first. Is that okay? But get our moderators out the door. I'm being bossy and just so grateful. What great presentations, right? Jeez. It is absolutely humbling that we're here. So I'm not going to talk, go eat, but bring it back because we're going to eat and talk because we're going to chat while we dine.
I know. It's okay. I'm good with that. Well, if,
there you go.
Janice, I think I see most people are eating. I think that's great. So this is still a big crowd, so thank you for sticking around. We have about an hour of time to discuss, you know, what we've heard today and hear suggestions and comments about where to go. But before we do that, we have the great fortune of having Peter Koch, Program Director at NIAMS, with us, and he's agreed to say a few words and maybe field a few questions. But I would like for you to remember that Peter is not in a position to change what's happening to the NIH. So let's be courteous to him. And by the way, many of you have made it a point in your presentations today. I think Corey started it, thanking the NIH for what they have done for their careers. I think this applies to many of us. And I just want to say that you talk to program officer and other officials at the NIH, and we're really fortunate that these colleagues are really trying to steady and get the ship continue to move under very difficult circumstances. So Peter, with that said, thank you so much. And then if you would say a few words.
Thank you so much. And just to build on what Pierre just said, if you want the hottest and latest news, I would suggest you get a Washington Post or New York Times subscription. A couple of things. First of all, thank you very much for having me. Rare diseases have always been a passion of mine. As somebody said many, many decades ago, rare diseases are, among other things, experiments of nature that give us the opportunity to help patients that suffer, but also provide great insight into very basic questions of biology across all of the disciplines. But you know all of this. So coming back to the NIH. So NIAMS, which is the institution that basically supports all of you, is a very small institute. So there are just 3 program directors that actually support the entire field of skin. It's Alexei Belkin and Anita Undala and me. So most of you probably still remember, well, will remember Riccardo Sibori. He retired a couple of months ago and he was replaced by Anita Undala. I think you can find on our webpage. And she's a trained immunologist and she has very extensive experience in running clinical trials. So if you're in the field of immunology, she's the perfect contact for you. So she worked at the NCI for a long time, so she has also a strong background in cancer in general. I think this is actually the most important thing. The other thing is, yes, we're living in a very changing environment. What I really would like you guys to do is just keep track of the announcement the NIH does. When I was a PI for 20 plus years, I never looked at any of those. Big mistake. These days, big mistakes. Read it, so stay informed. If you have any questions, any concerns, contact us. Sometimes we can't answer the question because it's above our pay grade, but we will do whatever we can to assist. I mean, our job is to make sure that you guys are successful. If you have success, we have success, right? So we're not the enemy, we are the friend. With that, any specific questions I can try to answer.
Any questions for Peter?
Yes, please.
Oh. Coming.
I have many patients enrolled in ongoing, not many, but a few, ongoing clinical trials. And I haven't heard from them recently if they're still able to travel to NIH. Do you know what's happening with those families?
You know what? Shoot me the email before I tell you something wrong. I actually double check. So I think as far as I know, this actually, the travel ban that existed quite a while ago has been lifted. It's my understanding they can, but shoot me an email. I make sure I give you the correct answer and double check. Even we can travel again. So we're here. So actually all three program officers are at the meeting. So if you want to talk to Alexei or Anita, find them or let me know and I'll make sure I connect you.
So one question that I could ask on behalf of the young people, Peter, is whether you could summarize what is a young investigator to do if there's an F31 with a diversity component or not. Should people expect that they may have to start over, or might there be an opportunity to revise the current grant proposal to address the concerns and the new directions? Can you comment a little bit on that in terms of if you're young and you're an F or a T or what to expect?
Well, you know, the diversity awards, right? They were judged by scientific quality as any other one. So I think basically if you were thinking about submitting one of those, I would say just submit to the parental grant and you were judged by the same people that judged you before. If your science was great and you were ending up with a fundable score, we'll do the same thing now.
Is there anything we can do to help you?
Well, so I think you help me if you help yourself. So, I mean, I'm a federal employee. I'm a representative of the executive branch. So I cannot talk to the executive branch what I think. That's not, they don't care. But you all are citizens, right? And most of you. So you can actually express the need for your society, for your research field, and for your research interests, and just make them known. I mean, you know, this is, people need to hear what the needs are. I mean, we communicate as good as we can, but then, you know, hearing it from the horse's mouth is the best way.
Thank you. That's precious. Ellie.
A question on behalf of the foreigners. So the question is, so I understand that there is now kind of a ban on having foreigners benefiting from NIH money, but does that also apply to subcontracts? So for example, if there is a technology which is available only outside the US, there is no NIH funded program which can benefit from that technology?
Well, so first of all, the issue is not with foreigners, right?
Yeah.
I was born a foreigner, right? But the thing is that the major grant mechanism that we're talking about, like our ones, they're institutional grants. I mean, they belong to the university, not you personally. That's different if it's a career award, right? So that's fine. No problem there. In terms of the foreign component, that has been paused. So it's my understanding that that the administration is working on a new mechanism that will be implemented. How they will look like, I don't know. I think they gave themselves a deadline of September to publish something. But I think for the time being, my impression is that you're encouraged to find your resources. That's as far as I can go. But I think everybody would agree, you know, if you read the announcement that down the line, you do not want to lose valuable scientific sources, and they are not always just around the corner. So there might be the other end of the world, right?
One thing that I would like to ask you about, Peter, is that, you know, if you look at this morning's symposium as a microcosm of what's going on in the rare disease space that is relevant to the skin. You know, we heard about many conditions, we heard about common mechanisms, we heard about cross usage of therapies for different diseases. So is it conceivable that NIAMS at some point could come up with a sort of a consortium type mechanism to get the first people, the PC people, the HS people, and Olmsted, I mean, to sort of come together under an umbrella and basically promote the sharing, so as an accelerator to solutions?
Yeah, so we actually have, I'm working on a program like this. I mean, this is a huge field and small steps is what we can expect. So this is certainly an interest of the Institute. Where the money will be in a couple of months, I cannot tell you, but I think, so there are a couple of interesting chances, but also challenges for us. So one is, if you think about rare diseases or any kind of, like, say, a cell or gene therapy, eventually to be effective, you will end up with technology that might be patented. Industry might be helpful if you are in the rare disease area, because I know people actually told me personally, so people that had patents and saying, we might consider giving you access to that technology if it's for a rare disease for academic purposes, not if you want to treat chronic diseases with a huge market. So I think this is the kind of question that I'm thinking about right now. So to open up opportunities and getting around these legal hurdles that we all have, the restrictions and what technology we can freely use and we cannot. But certainly, I mean, this is an interest of mine, and the Institute has been very supportive of that idea. But you have to find the money.
Yeah. Oh, question on Zoom. Just wait a minute.
Hi. Thank you very much for taking questions. So we heard a lot of talks this morning that illustrated the complementarity between studies in animal models and studies in human subjects. And what I'm wondering is, given the announcement from the NIH last week or the week before that preference will be given to studies that have a human component, what's your understanding of how absolute that preference is likely to be? In other words, is it going to be the case that all studies are going to need to have some human component to them? Or is that just going to be a population level shift in preference?
Well, so you're asking me to read the tea leaves, right? So my guess, and this is my personal guess and not an institute statement, is that we always talk about the gold standard of science. So I think what's the appropriate, scientifically appropriate approach will always be acceptable. At least I very much hope so. It's not that people will tell you ahead of time what type of models you use for your research. I think that that is not how we have operated science, but you will see how the future will develop. There's no strict guidance at this point that would prevent you from doing animal models. I mean, we have a big program that works on the effect of toxic chemicals on skin. That's the CCRP. This is an example where you actually, at least until now, you have to have certain animal models that are closer in humans, like mini pigs, to actually prove efficiency of countermeasures to block the effects of toxic chemicals on the skin. So I think that's an example where, you know, I think the science should always be the part that actually determines the methodology and not the other way around.
Thank you.
But that's just me.
Thank you, Mike, for still being with us without a lunch. We appreciate it.
We don't know. It might be there.
It might be better than ours.
Any more questions for Peter?
If not, then thanks for having me.
Yeah.
You know, we're just trying to get a good relationship with NIAMS and also NCATS. And I just wanted to just give a shout out to Peter, because when I first went to DC, I asked if I could meet with him and he said yes. And he spent an hour with me, just a regular old Yahoo like me. And he's been really supportive ever since. So I just want to say, as a patient advocacy organization person, thank you.
I would like to disagree with your self-characterization as a Yahoo.
Oh,
You're phenomenal, Janice. Yeah. Okay, so now let's talk a little bit about the science related to PC, what we've heard this morning that might have struck a chord with you on any front, whether it's in, the lab research, company-driven research, clinical aspects, the new classifications. Now you've all had a chance to think about it a little bit. So let's open up. And I think that maybe we'll have a first question from our first topic from Robin, won't we? And then I'll pass this over to Ellie.
Well, so there are a few questions that we thought of beforehand, and one of them is, what's the most promising pathway forward for PC at this point? And I would say my question is for PC therapeutics at this point. And so we could, just based on what you've heard today, or based on just your knowledge of PC over the last 20 years, where do we think we are with PC therapeutics? Where should PC Project be putting its effort in pushing these forward?
Thanks. I'll just put in a big plug for drug repurposing. I think there's huge treasure trove of drugs that have been shown to be safe and effective for other diseases that you don't have to start at phase 1 in animal studies and stuff. That I think there are things that are out there that really could help. So I don't know how PC could fund more widespread searches for those types of things, because they're not generally funded by NIH as far as, screening types of things, but I really think that could be helpful. It's just you need a good assay that's high throughput. So if there's some way to do pilot awards or something like that, I think that really accelerates the from the lab to the patient timeline. That could be really helpful for patients. I'm really excited about gene therapy, as I mentioned, but there's a lot of development in those products. They may have to be commercial areas adapted for each patient, but I think drug repurposing is like a huge, huge untapped research at this point. And people have done it. Dr. Sprecker's done it with this ruxolitinib, and we're trying to do it with MEK inhibitors. I mean, I think it really could be powerful for your community in general.
So Ellie, would you like to say more about drug repurposing? Because I know that, so Erwin McLean's group they used the K6A promoter, looking at linked to Firefly luciferase, and used that as the screen to look for compounds in a repurposed library, for upregulation or downregulation of K6A, and they did find the statin drug, and then there was a little bit of work done on statins, but I don't think much has been done since then. The work that Roger Casper did at Transderm wasn't necessarily, well, that was with rapamycin, but that wasn't necessarily from a library screen. That was just based on a hunch that rapamycin could potentially downregulate expression of K6A. So where are we with screens?
So first of all, I think that I would like to take the question to somewhere else, not and leave the scientific arena and move more to a kind of a strategic kind of view of how to deal with repurposing of drugs. The major problem of repurposing drugs for rare diseases is that there is very little money with that. So how do you bring the industry, because it doesn't matter if it's a new API, if it's in an old drug, to do a properly designed clinical trial will cost a lot of money. And we need a placebo control, or at least a clinical trial with a standard of care comparator. And here we don't have any, so it will most probably be a placebo controlled clinical trial, and this will cost a lot of money. So we need the industry money. On the other hand, we are not a market for anybody. So the question is, how do you do that? And there are many ideas that could be brought up. I mean, I have a suggestion, but I would like also, of course, to hear if you have other ideas as well. So I think that there are many people in this audience and other which are also dealing with rare diseases, but are also a member of larger societies dealing with diseases which are of great interest to the industry, like psoriasis. We heard a little bit about atopic dermatitis. There is HS. There is a lot of overlap between those rare diseases and much more common diseases. And something that could be done at the level of the organizations, for example, patient support groups for rare diseases versus larger organization, is to kind of form an alliances. And those alliances could include conditioning the participation to those large trials, because the industry is very much dependent upon the clinicians to somehow give a bit of the benefit from those large trials also for rare diseases. But this is a kind of a strategy which has been actually put in place, I know, for very rare arrhythmias in the cardiology field, but maybe could also be relevant to us. And this is something that should be discussed at higher levels. But this is the only good idea that I've had in order to try to bring those repurposed drug trial to the level of sufficient evidence, because at the end of the day, we have to come back to the patient, and they don't care if they are affected by a rare or a common disease. They need strong evidence to make sure that the drug they are going to take, with all the side effects associated with it, is really going to benefit them. And there is only one way to give them an answer to that question, and it is through data collected to properly conducted trials. So that's my idea, but maybe there are additional ideas. Kate.
Well, I'm sorry, I've got the microphone, but so one of the industries that I think that we should be looking at, and I think would be potentially very interested in partnering with respect to repurposing, is the large language model AI people, right? Because what they're starting to do is look at how you predict whether or not that using large language models, how you predict binding and effective repurposing, right? And we have like one of the, these are that we have more knowledge about what the target is in these rare disorders than anybody else. And so I think that would be potentially a really good partnership for us to be able to really get into and make a very good case for how we would be able to identify things. And then they go out and they license or whatever they have to do. I mean, a lot of the molecules, they don't even have to, because they're just out there being used already anyway, by definition of repurpose. So I think we should look into finding those companies that are really interested in a proof of principle, and we should be one of their first proof of principles.
So I wanted to follow up on this question of rigor, because you mentioned the need to do randomized control trials to really understand whether medications work. And yet in dermatology, at least in the US, there's massive off-label use of medications. And the only publications that come out are when the drug worked, but not when it didn't. And so I'd like to raise the question of whether patient advocacy groups could have a role in reporting both the good and the bad. So to really ask that all patients who are trialed on an off-label medication actually have a standardized reporting form that comes back to the organization. I mean, PC Project has been so incredibly organized in the way that it's approached phenotyping of patients. I think the next stage, and FIRST could do this as well, would be to really say, what's the evidence in a large number of patients that this has effect, even if it's not a randomized control?
But I would argue that if we base our recommendation upon anecdotes, and it doesn't matter if those are positive or negative anecdotes, we may either adopt drugs that do not work, or actually give up drugs which have a huge potential based on that. So there is, I'm not sure that there is an alternative to well-conducted, costly, time-consuming, resource-consuming clinical trials.
This is Joyce. I'm a pediatric dermatologist, so I want to put in a plug for drug repurposing as well. As presented this morning, as you can see, many of the concept of drug repurposing has been developed from a really solid science. Animal models, in vitro studies, and mechanistically, they work. And I fully recognize it, especially for PEDS patient, a rare disease, start in the congenital stage and presented very early on. When we have these new molecules and new treatments, it takes years before it's really coming down to the pediatric population, the population that's really deeply affected. So there's a huge advantage for congenital rare genetic disorders. We look at the recent call from NIH about the CORI outcome for rare genetic diseases, and looking at these therapy, comparison of the therapy. And some of these neurotherapy cost millions, many millions. And, I myself, as an investigator, clinical investigator, has benefited from government funding, especially FDA orphan disease programs, and receive funding for using some of these repurposed drug for rare genetic conditions. So I really think that there's less industry interest in these old drug, but clinically they'll benefit our patient and get into clinical care much faster, can be fast tracked. And from just our society, healthcare delivery system, from long-term point of view, we got to conserve our resources somehow, and we can't afford these super expensive drugs. And whether there's a way to expand support from FDA or other programs to support drug repurposing.
Thank you. So maybe I'll bring up another issue. So yesterday we had a very good discussion among the EMSA people, and there was one topic which I think was again somehow connected to clinical trials and in general human research. PC is a very rare disease, and at this moment, for example, there are 3 different clinical trials which are ascertaining the efficacy of ruxolitinib in PC, one in France, one in the UK, and one in the US. There is no harmonization between the protocols used in the 3 countries. So when the results will be available, it will be very difficult to combine them and to increase the statistical power of the data. So if you study hypertension or asthma, it's not a big problem. But if you study a disease as rare as PC, it's more of a problem. And so we were thinking that maybe one of the future tasks of our group would be to kind of put together protocols, which then could be used everywhere, be made available to everybody, and then could be used everywhere. But at the end of the day, we would be able to then mix or combine the data and somehow get some more meaningful results. Peter.
I thought that was a very interesting thought, and I have one that's seems like completely unrelated, but is a similar area, and that is in the area of stem cell therapy. There's the same problem. People in different parts of the world develop protocols, different approaches, slightly different, and the products they develop are necessarily compatible try them in humans. So what the International Society for Stem Cell Research right now is they basically set up groups that basically develop harmonized protocols for conducting certain types of procedures. And actually, in their case, they even adjusted them to the legal frameworks in the different countries the experiments are done to make sure that they can be applied there, can be approved there, and can be compared to other countries. I think that's a very essential part, not waste resources and expose patients to unnecessarily risk.
Thank you.
One of the reasons that we have to think a little bit about how we're going to be deploying PC project resources in the coming year to support research would be, we're thinking about a new program, not a big program, but incentivizing people to share as they're developing protocols. So by seeding it with a little bit of money, incentivize people to communicate, to be in touch with one another. Is that something that you think you would respond to favorably? I mean, would that be a good type of investment on the part of the PC project? Just, I think that, you know, we cannot write your protocols. I mean, we wouldn't want to do that. Maybe in some cases we could. But an alternative would be to have a program that incentivizes people to, you know, engage one another across states, across countries, to try and harmonize so that there could be combination of data. Does that sound attractive, people? If PC were to try and incentivize that? Janice, are you gonna kill me for saying so?
The idea is to fund small groups of people who would get together and develop those harmonized protocols. And then once the protocol has been well established, then it would be shared with the community, and everybody who is interested in doing PC research would agree in advance to adhere to that protocol.
Trial design.
Yes, exactly.
I mean, I was in the EBSAB, but I, you know,
Exactly.
yeah, so
Would include everything like the power calculation and everything in order to make it.
including endpoints
valid.
and things like that.
Exactly. We will get to the endpoint in a minute.
Oh, yeah.
Oh, yeah.
Yeah, I think, you know, in theory, it could be a good idea, right? You're coming, you know, as we're coming into this from the industry perspective, right? You're left to kind of design these clinical trials. You don't have a ton of information around them. And so there's probably quite a few elements within a clinical trial protocol where that could be beneficial. And I said, we'll get to clinical outcomes because that's going to be the big one that's going to be difficult to harmonize across, because you may have different drugs affecting, you know, different pathways, different, you know, manifestations of the disease. Like one might treat hyperkeratosis, one might treat pain, et cetera, et cetera. And so getting good validated clinical endpoints that we've done, and I mean validated as in we follow the FDA's process for validating clinical outcomes, right? So we need to do it that way. And if that's the case and you have those outcomes validated, you're not going to just have 2 pharma companies, you'll have 50. Okay, if you have really good outcome ways to measure PC.
So, but the idea, of course, is to have one protocol per one experiment. So it's a different protocol for each and every drug. The thing is that the outcome measure of that's what we understood yesterday, of at least 2 of the existing protocols regarding ruxolitinib are looking at different outcomes, which was at least at different primary endpoints, which is really very disturbing.
Can I see a show of hands? How many people here have actually run a clinical trial? So about maybe 40%. How many people have run a trial in PC? About 5. So I think that absolutely this is needed, because in the PC trial with topical rapamycin, when the endpoints for pain, it's really challenging. It was really challenging to capture the improvement patients were making. And it is so complex designing clinical trials and understanding how to get the right patient-reported outcomes, which are becoming more and more important in understanding therapies and how they're impacting patients' lives. We talk about quality of life in general, but quality of life measures are becoming essential differentiating medicines in trials. And it is not easy to pick between them. And so I really do think that this is a good idea, that we should be, as a community, working together, because we can't afford more failed trials, or at least from a design standpoint, right? If we have a great medicine or a medicine we think is great and it doesn't work, that's fine. But it shouldn't fail because we as a community did not put our heads together and come up with a better design. And from the first perspective, or the ichthyosis perspective with topical isotretinoin, this medicine, topical isotretinoin with the polyethylene glycol vehicle that they developed, it did work. 70% of the ichthyosis patients reported improvement. The problem is it was in the drug and the placebo arm because the polyethylene glycol was active and actually benefited the patients. Had there been a third arm, or let's just say a third arm that was just current over-the-counter emollient standard of care, we might actually have a drug that's going to be approved for ichthyosis patients. And this was something that wasn't anticipated, and maybe with a collective minds of people that had experience in ichthyosis, we could have hopefully foreseen this. But the thing is, the drug did work. The patients, every single, I think Joyce, you were in that trial. Every single patient was begging me, "Where's the leftover medicine? I want it. This is the best thing I've ever used." It was a phenomenal outcome, but the trial design failed us. So I really do believe that we together should come together. And the last part that I was going to make, Pierre asked me to talk a little bit about academic industry collaboration. I'm a big believer that we can't do this alone. We can't help get drugs to our rare genetic disease patients alone in academia. We have to partner with industry. That's my personal belief, because there's just so much that goes into bringing a drug in, to the market. To me, my opinion, academic industry, it's all about relationships, personal relationships. And I know there's several people in this room that have personal relationships in industry. And I think we ought to coordinate and grow those relationships to make them see the value of rare genetic disease. One area where that I have heard of, so I'll just give my anecdote because I think it explains the potential that we have as a community. So first, when topical isotretinoin, which was the timber drug for ichthyosis, it was purchased by Leo Pharma. Leo Pharma wanted to get into this space. They wanted to help ichthyosis patients. They were excited about it. And then the drug didn't pass phase 3. Industry is willing and able. And as soon as Leo backed out, or they announced the failure of their drug in phase 3, I had 2 companies approach me. We're ready to fund the next ichthyosis trials. 2 companies, bam, like that. They are interested. They just need the community to rally together and push them over the edge. And the last thing that I'm going to say is one thing that I don't think PC Project or FIRST has done a very good job of, but we have to do now, is underlying the inflammatory nature of these diseases. Because what pharma is interested in is inflammatory diseases right now. And we need to show them that this isn't just a rare genetic disease. People have pain, people can't walk well, people have scaly skin. We need to show them the inflammatory nature of these diseases that goes along with everything the patients are experiencing, because that, in my opinion, will get industry's attention. Talk to me after. Well, actually, no, in seriousness, what I would do is I would get all of the leaders here and let's put together a consensus paper about inflammation in PC, inflammation in ichthyoses. Let's actually start by putting together a consensus paper that talks about the inflammatory nature of these diseases.
There are also some clinical trials that are ongoing for rare genetic diseases using biologics, so for instance, some other medications. So we need to kind of promote the concept, probably discuss and talk about it a little more.
So I wanted to say that like the definition of what inflammatory disease is, I think we probably need to expand. So I think a lot of the science that we've heard, Pierre's work and some of the stuff that I didn't get into yet, but there are inflammatory pathways that keratinocytes are very active participants in, and I think that's under-recognized, but is becoming more recognized. So it doesn't have to be that everything is in, IL-17, IL, whatever we have a hammer for kind of thing. But I do think that there are common inflammatory pathways that we could target, and that's why I was excited to hear Pierre's work and talk to him about JAK inhibitors when I first heard your presentation of that data. I think that could honestly work, even if it's not necessarily a systemic thing, but if it's a keratinocyte-induced inflammation, I think that's very important. I wanted to talk also, maybe you're going to get to this later about outcome measures, but one thing we've struggled with, if we're trying to learn from other rare diseases in the pemphigus and pemphigoid group, which are autoimmune diseases, is that the outcome measures can really tank a trial when it goes to FDA for approval if they're requiring you to do an IGA, an investigator global assessment. That can really sort of derail something that looks very promising if it wasn't set up to do the type of measures that are now being prioritized, which is this sort of simple 0 through 4 grading system that other countries don't necessarily use. But I would get the FDA involved early on so that you know, where do we need to pitch our outcome measures to make sure that every body, every different decision body can agree with accepting our measure before you put the money in, do the trial, and then realize that you didn't get a 2-point change in the IGA and now you can't get an approval. That's devastating to a rare disease community that puts a lot of stock in a drug and it shows promise, but the outcome measure wasn't designed right. So I would definitely encourage that.
I was going to ask Jan a question. So with the idea of PC Project facilitating the design of these trials, so in your experience, have you had pushback, or have the industry collaborators, partners that you've worked with, have they been pretty good at taking advice from PC Project about what endpoints to use, or they have their own and they're moving forward with their own?
I would say yes and yes.
Okay.
I think I've really appreciated the industry partners that have wanted to have focus groups with us, you know, and to say what's important, what's meaningful. And so, yes, we've had a lot of people ask us questions. I've been on Zoom calls, even with people in this room, and even with the Palvella study. I know they get knocked a little bit sometimes, but you know what? They converse with, and our team lots of times. And so I feel like it's important that we are heard, you know, and we know, and we also even know how tricky our disease is. We're weird. I mean, we're weird when it comes to evaluating us because of the severity spectrum. And we clearly have a high placebo problem. But anyway, so yes.
Thanks, Jen. I'm gonna hand the mic to Pierre, but there is Michael Caterina has a question on Zoom.
Hi, yeah, I wanted to say something about the concept of outcome measures, and particularly pain as an outcome measure. You know, as I'm sure many of you appreciate, it's a very difficult outcome measure to standardize in most studies, particularly in studies where pain may be of various natures, come from all over the body and so forth. But I think there are some advantages, if you will, to the focal nature of at least the palmoplantar pain in PC patients and other patients with painful PPK that might be exploited in a way in these clinical trials. And what I mean by that is that one might think about devising very structured activities that the patients can engage in on, say, a daily basis, and subjectively evaluate their pain experience during those very structured activities. I'm thinking of things like standing barefoot on a textured surface, or having a certain weight on the palm of one's hand. You know, the fact that much of the pain is localized to these sites, and that we have the advantage of a very engaged population of individuals who are living with PC and these other conditions, might allow that kind of serial assessment to reduce some of the variability in the readout. And granted, as we heard earlier, any readout is going to have to pass the muster of the FDA and other regulatory authorities. But I can envision that one could craft these much more standardized readouts to be able to get pain variables or pain measures that one can look at over time during the course of a clinical trial.
I think that combining your comment with something that Sansi said a bit earlier with wearables might be a way to go.
That's one option. I want to say one of the recent times we've seen that actually work was in the Abiona trial, where they would actually link the wound healing to pain during a bandage change, right? So it was a very specific type of pain and a very specific type of event. And I think if we can figure out what that event should be, or what that activity should be for PC, then you may be able to narrow in on a pain score that would be usable.
I think that we ought to look at some of the other trials going on in conditions with pain and understand how they dealt with it. For example, we could look at the sickle cell disease community, and also Disc Medicine is developing small, or has starting a phase 3 trial on a small molecule to treat certain types of porphyria, where there's a lot of pain when the patients are out in sunlight. So some of the ways in which they're developing strategies to deal with pain and activity change is actually very effective. They've done very well in their phase 2 trial. And I think that drawing on what other pain-related diseases have done is very important. We can't just stay isolated. We got to see the big picture of what other people have done. And I'll say to Corey's point about the inflammation is we've known this for a while. So our group, led by Minho there in the back, we published 2 years ago a paper, the largest cohort of Southeast Asian ichthyosis patients, and showed that many subtypes have upregulated JAK-STAT signaling. So this inflammatory nature and the ability to target some of our rare genetic diseases with JAKs is old news.
So I've asked Irv Epstein, who has a long record of making discoveries, to make a few comments.
Well, my most current discovery is that this is the best part of the SID meeting last year, and I'm sure this year as well. So I really congratulate. The science is remarkable, and I have all sorts of notes saying, how can I apply it to my own interests? So for more than a half century, for reasons I don't really remember, I've been interested in Gorlin syndrome. For those of you who don't know, it has many different phenotypic abnormalities, but for today, most prominent is the development of lots and lots of basal cell carcinomas. So basal cell carcinoma is the most common cancer of human beings. In some places, half of all cancers are basal cell carcinomas. So one advantage that we've had, so instead of getting 1 or 2, if you're of European descent and at age 50 or 60 or 70, these people start getting them at age 20 or 15 or even 5 sometimes. And it can be devastating because once you've had 200 Mohs surgeries, you really don't want to have any more, for God's sakes. And 30 years ago, it was identified by our lab and the whole rest of the global village simultaneously that there's a specific gene mutation which causes upregulation of the hedgehog signaling pathway. And that's the cause of most of the cases. And probably all the cases, even if they don't have a mutation in that gene, have a mutation that upregulates hedgehog signaling. So there's a lot of overlap. About a dozen years ago, 2 friends and I, Gene Tang, who some of you may know, and a guy named Phil Beachy at Stanford, were sitting around having coffee and patting ourselves on the back because Genentech had taken the information from the mid-90s and developed Aravage, a topical hedgehog inhibitor that certainly was not gene therapy, but it blocked excessive hedgehog signaling. You can take a patient with 100 basal cell carcinoma and give them 1 pill a day, and after they have an 80% chance that they'll have 0 basal cell carcinomas at the end of a year of 1 pill. And if you do a biopsy where there was a pill, where there was a BCC, it looks completely normal. Problem is when you stop it, they tend to come back. Well, why would anybody stop it? Because no surprise, there's side effects. And so about 13, 14 years ago, the 3 of us were sitting around saying, well, isn't there some other way to use the genetic information that we know, the molecular information, and convert it into something that might not have quite the same benefit, but would have some benefit and not quite the same adverse event. So being the 2 of the 3 of us are dermatologists, well, rub the stuff on. So what do you do if you're in the Stanford environment? You have an idea like that. Start a company. Now that may or may not have been a good idea, not sure. But it is, what I am sure about is that it takes an absolutely obscene amount of money. Obscene. And we're now doing a second phase 3 trial because it worked in a subset beautifully, but didn't only enroll patients with that subset. I can talk to you about the details. Probably not. And so it is very expensive, obscene. I think if this drug gets to market, which I think it has a pretty good chance of doing, it will have been $1/3 of $1 billion investment. Not all of it by us. It's now on the 4th company that's been trying, and the first 3 have gone bankrupt or have given up or whatever. So it is just mind-boggling. It's for somebody who's in private practice and thinking about $12.95 for freezing a wart. $1/3 of $1 billion is pretty different. You don't have much experience with that. But I think, first of all, the fact that rare diseases will get a higher recompense per treatment, per patient, is stimulating to the industry. And certainly that should apply here as well. And secondly, the chance that maybe you'll learn something from this exercise that will go over to the 3 million people with basal cell carcinomas that are sporadic and appear in the United States and a similar amount in Europe every year. So one thing that comes to mind sitting here is there are a lot of people out there who get blisters and calluses. How can you say that what you're doing here for this rare disease will do something to make the people who are out there playing tennis on weekends not get calluses and blisters? Or the Defense Department should certainly be interested in that because people are still marching along in boots. How can you do that and sell it to them on that basis? Get them to understand. So that's one thing I think I can say that would be applicable. Second thing is that, you know, about basket trials in breast cancer in the first place. And the idea is that there are plenty of drugs out there, but there aren't many patients. And setting up a trial and gathering rare patients from around the world is a very expensive proposition. So the Scleroderma Research Foundation set up a process where you could have a basket trial in scleroderma. And what that means is they have, I think, 140 sites around the country, around the world, who have agreed to participate and enroll scleroderma patients in trials. And so there's a common protocol, there's a common endpoint, and there's one common placebo group. So the one placebo group is good for all different kinds of drugs. And the hope is that if you have the mechanisms, the infrastructure to do a clinical trial, and this group could have that if you collaborated, which is why I said maybe it's not such a good idea to set up. There may be lots of drugs that are out there that could be repurposed for this disease. So I would think about that as something that Janice in her spare time might set up. But I think that there you might, you know, if you build it, it'll come. Maybe the industry would give you then the money to run the trial because it'd be a hell of a lot less expensive and less, but not only expensive in output, but expensive in their people who would have to be hired to do the trial, to oversee the trial. And maybe that would be something that could be done in this rare diseases. And I'm not sure I just said it, but this is why I commented earlier. Maybe it wasn't such a good idea to form a company. Maybe what we should have done is get together with other people interested in Gorlin syndrome and start a basket trial that we could offer to people. 2 ideas. Thank you, Pierre.
So we're just about at the end of the half hour, and I'll let you say something.
No,
I
I'm
just
not
want.
saying anything. This guy's kind of twitched a lot.
Oh, I see. Great.
All right, thank you. I'm new to the PC field. I study DNA replication and cancer. In fact, I have a skin cancer drug in clinical phase 2, which will be presented on Friday poster. So my question to the experts in the field is that has anti-cancer drug or similar anti-proliferation drug been tried for PC? Because our drug works for psoriasis as well. I was there in this morning.
So wait until what I said works. months, they'll have a whole portion fixed up that you can pay them to do the job.
So one thing that I want to say before we part ways, I think it was a very successful event. I just want to say thank you to the session chairs and most especially to the speakers and most especially to all of you for sticking around and being so engaged. So thank you, everybody. So any kind of, in the last few minutes, I think that one. Good, good.
Actually, we do. Yeah, actually, we do that very frequently. For example, I'll give you two examples. And a lot of the hot mutation occurs in PI3 kinase, and, like a brain cancer, you know, you name it, and many different type of oncological diseases. And so one of the PI3 kinase inhibitor from Novartis, alpelisib is the generic name, is frequently used for different type of a cancer. And now it's just received a provisional approval from the FDA for children with vascular anomaly. They're hyperproliferative, and they actually give provisional approval starting at 2 years of age because there are no therapeutic options for these patients, and it fast-tracked very, very quickly. And we've been using that for the last 2+ years, and it's really beneficial. And another example, as you heard from this morning, we talked a lot about the activation of MAP kinase and ERK pathway in some of these keratinopathy diseases. And now, you know, trametinib, as you know, is FDA approved for melanoma. And so as several other inhibitors that Dr. Simpson listed, the BRAF inhibitors for many of the different oncological disorders. But these targeted therapy, absolutely, now as we understand the underlying pathogenesis and mechanistic, you know, mechanism of action, we are constantly thinking about how to repurpose these drugs for other non-oncological disorders.
One struggle, I think, is how the drugs are housed within an industry that's a large pharmaceutical company. So like dermatitis is owned by Novartis, and they tend to group oncology very separately from their skin arm. And there's not actually a lot of talking that goes on between those, as I found out where I was trying to get in to talk to someone about, could we get an investigator-initiated trial for this thing? And it was like, you know, knocking on just death's door. It's just like, it doesn't seem to resonate sometimes when industry, that larger companies are siloed. So I'm interested to learn, like, how do you break through those barriers between like an oncologic indication versus something that could be totally different and repurposed for a disease that nobody would necessarily connect with, using a treatment for melanoma to treat PC or Darier disease or whatever. So I think that's a little bit challenging with the way that some companies are organized. Not all the right people are talking to each other.
Yeah, that's absolutely true. It's very hard to even get people to talk to you about dermatologic drugs at certain companies with certain pipelines. I just wanted to real quick just bring up, many of you may be aware of the fact that the rare pediatric disease voucher is set to sunset in September of 2026. And I bring this up because not only, you know, do we are retaining everything we have in the Orphan Drug Act, of course, but these vouchers have been kind of essential, right, to encouraging companies to study pediatric diseases, to repurpose drugs into pediatric diseases. And you know, there's a lot of concern that a lot of that research will dry up, a lot of the investment will dry up. I know when you know, personally, when we go out and we talk to investors, it's a really important thing that we bring up. And it really, on the long term, really doesn't cost the taxpayers a lot of money, right? This is not a tax heavy thing. But it really is important to corporations, especially when you're trying to do the whole risk benefit reward analysis that goes into Where are we going to invest the dollars that we are able to raise from investors? So I just want to point that out. In addition to, I know everybody has other problems with the NIH, but this is another one that we really want to get it through Congress and maintain those pipeline of drugs. So thank you.
Yeah, so I have a science-related question. So I was quite intrigued by Benjamin's talk. We talk about contractility. So if I think about contractility, is really to think about the actins. Then, so I think we focus on keratins for a reason. Then keratins interact with the desmosomes. Then, so we have expertise in this room. Actually, Cassie Green's group also published linked desmoglein 1 with keratins. They have physical interactions. So, we're talking about the intermediate filaments. So how about other parts of the cytokeleton, other crosstalks? So are we talking about the cytokeleton related disorders? PC is just part of it. So there are so many inhibitors, activators along microtubules and actins. So is this opportunity for us?
So I guess, I think there's a lot of literature on interactions between different cytoskeletons. And I would hypothesize that the reason we are here talking about a disease caused by mutations in keratin genes is because there are so many of them with such specific expression patterns that there's a lot of potential for compensation, and these mutations are not lethal. I think if you have disruption, significant disruption of actin or microtubules, there's a lot less redundancy. There is isoform variation, but not nearly as much as intermediate filaments. And I think we don't have diseases, we don't know of diseases caused by or closely related to those cytoskeletons because there is less variation and it's would be lethal.
I would say also, we like to draw diagrams in textbooks where one cadherin binds to one catenin binds to one cytoskeleton, because that's a simple way to teach cell biology. But I think there's a lot more interaction that happens between these complex junctions at the cell surface to actually form the physiologic junctions that actually hold cells together. So come to Jess's plenary talk this afternoon, we're going to talk about how a desmosome defect actually seems to cause an actin problem. And Kathy's group also, with a lot of others, has done some profiling of biopsies of patients who have a quote unquote desmosome disorder. But one of the main signatures is an actin disruption. The Rho-A pathway is a major regulator. There are targetable things in that pathway. That happens to be one of them. But I think there really are convergence on these pathways that sense and respond to mechanical stress that may affect the way a keratinocyte behaves, adheres to another cell, whether it can close a wound or not. And I think the biology there is super interesting, but mostly because it's going to be therapeutically actionable, I hope.
Certainly. You guys have the expertise, through the agonists, antagonists, on the existing models and see what happened. Appreciate it.
Ongoing.
Okay, thank you. Wow. You guys did exactly what a consortium is supposed to do. Collaborate, talk together. I thought it was, you guys like this? We've never done this before. Should we do it again? I mean, did you like the trapped lunch? I remember we had a dinner cruise in Chicago one year and Pierre's like, oh, you trapped us, so we have to talk to each other. And we said, yes, we did. Anyway, enough said. You guys are fabulous. I love the discussion. I was back there typing, taking notes. Just thank you. I hope the collaboration continues. And I appreciate Pierre looking out for us. As I announced earlier, we're looking at next year doing something with the Haley Derrier group and the ichthyosis to kind of work on what we have in common. And yet Pierre, bless his heart, said, let's protect the PC space because we've been planning for it for a long time. And yet we can clearly see, I mean, we're bringing Diana, we're like, itch and pain, itch and pain. I mean, we're all about that, right? And so I invite everybody to go forth as you go to the SID, look for possibilities here and come back, talk to me, because you know what? I'm not very smart, but I work with really smart people, right? And so keep collaborating, but most of all, thank you so much. We're rare. And honestly, sometimes we felt like we get left behind a little bit, but we don't feel left behind in this group. We feel like there's people that absolutely give us their hearts, and they're all of you, and you care for us. So go forth, have fun, but remember, we're right here and your family. And if you came in and you weren't registered, my husband got you registered, so you're part of us now. So sorry. So we'll see you. Watch for our newsletters. We'll tell you what to do with the grants, right? We'll figure it out, get it right, and let's stay in touch. Take care and thank you. Oh, the forms. Thank you. I want your forms. And if you look on the brochure and you see info@pachyonychia.org, you can all answer that email too if you have lots of ideas. Give me the forms.
Yeah, I agree with her. It's the best part. You did a great job.
We did a great job.
Okay.
We did a great job.
I'll take some of it, but you did a
You
great
can
job.
take all of it. Goodbye, everybody.
Okay.